they can't. It's shared with them. So
okay.
the screen is shared.
Okay, great.
And they also see the video, so they have both video and screen. So, yep. Because I can see the screen here too on mine that's being shared.
Hardy, Jim, Danette.
Hello.
And one of the other boys.
All right. Well, let's get the show on the road here.
That's Jack.
Jack. Second. I'm going to close mine so that I don't.
Camera's there.
This is predict the cameras on.
Okay. We've still got a couple of minutes, but
Just go ahead and maximize that again.
yeah, I think that you're fine. We've got time. All right. Well, I want to welcome everyone here to this colloquium in honor of Dr. R. James Hickey. We call this questioning species and species concepts, because I think it's the thing that reminds me the most about interacting with Jim was this constant questioning of ideas, and is that a species? Is that a real species? And one of the great things I learned was this sort of incisiveness of asking those hard questions and really pushing you and wanting you to have the data and evidence to defend those ideas and boundaries. And so we've convened a number of folks from the Hickey lab over the years to speak about their current research and maybe retell some of their stories about Jim. Jim and company are on video link. Most of you in the room know this already, but for those of you that are filtering in still, Jim's on a video link coming from Oxford with Hardy and Jack Keegan and a few other folks in the room. So if you say any bad things, he can hear you, and that may be not good for you. But anyway, I'll let that go. But anyway, to start us off today is Jim's longtime colleague and collaborator at Miami University, Dr. Mike Vincent, who's going to introduce us to Jim Hickey, the development of a scientist. All right.
Thank you, Mike. All right. Hi, Jim.
Hello, Mike.
How are you?
I'm doing great.
Great. Hi, Melanie. All right, she's there too. All right, so what I'm going to do today is just sort of run through using sort of Jim as an example of how people develop into scientists. And then he's going to be my model organism today as we're talking about his development as a scientist. And I'm really just going to run through Jim's life a little bit and talk about it and introduce you to my friend. So if you have ever been to Jim's lab or his office, on the door, there is a lineage there showing Jim Hickey being descended from Linnaeus. And that is something, I'm sure he's proud of the fact that he can trace his academic lineage back to Linnaeus. But that's only one aspect of what Jim's development has entailed, is that academic lineage, which is a great one and something to be proud of. But Jim was born in 1950, 4th of September in Brighton, Mass. He was the son of Ralph Burgess Hickey and Doris Lapham Hickey. And if anybody has ever traveled with Jim, you will be familiar with that head on the arm like that, because when he's asleep at night sometimes, he sleeps like this. And the first time I ever saw that, it freaked me out pretty bad. But that's Jim at a young age. Now here's a picture that I think is really telling. You can't really see it very well. It's from a newspaper or an old photocopy or something. This is Jim as a young lad with an eastern garter snake at Miles Standish State Park in Massachusetts. Keep that image in your mind as we go through this talk, Jim and snakes, okay? Pay attention. Here's a picture of Jim with a bunch of other campers delighting over a raccoon at a young age. And again, that's from a newspaper article, so the quality is not really great. But Jim has always been interested in natural history. And so that's a really great picture to show that kind of interest that he has. So Jim attended Brighton High School in Massachusetts. And on this side, the right over here, you can see Jim the Suave. And there he is in his high school yearbook. I told him I was like the FBI. I could ferret out all the pictures, and so I've got some of them here. But I want you to look what's written in his yearbook, that he wants to be a biologist, and I think he succeeded at being a biologist. And there's something else down there that I want to point out to you too, and that's the quote down there below where it says his favorite pastime is scuba diving, which was news to me. Look at the quote that's down there at the bottom. Two qualities that often blend, a good sport and a loyal friend. And I'm going to come back to that later. When Jim went to college then, he went to Bridgewater State College in Massachusetts and studied biology and got his degree in 1972. Here's a picture of Jim doing photography somewhere during that time period. But there was someone there who was a Miami grad, who was a botany teacher at that institution, whose name was Walter Hewitson, and he had a really great impact on Jim. Jim said that one time he asked him if he was stupid or just lazy. And so, you know, that changed his life, I suspect, a little bit, you know. And he had a great impact on Jim. And Jim's teaching style especially, I think, was influenced by him. And Jim said that one of the things that Walt Hewitson said to him was that the key to success is to always be yourself. And I think Jim has always been himself in the classroom. All right, here's another person that was very influential in Jim's life, and he met him after he graduated from Bridgewater, and he was thinking about going to college. He went to the botany meetings that were in Amherst, and he met David Bierhorst, who was a morphologist, and this man also really impacted Jim in the way he thought about the organisms he was looking at and the science that he was doing. And so this is another person that I think was really influential with him. Now, if you don't know, on the whichever hand this is, the right over there, that's Isoetes. That's a plate from the Bierhorst morphology book showing some anatomical and morphological features of Isoetes. Now, eventually, Jim ended up at Miami University, where he studied for a master's degree with Hardy Eschball, and two people especially at Miami were really influential in his development as a scientist. Hardy Eschball, who was Jim's advisor, and T.K. Wilson, who was an instructor who really challenged all of us. But just a fine teacher and a good friend. Both of them then really were impactful in Jim's development. Jim did eventually finish his degree. He got a master's degree studying the variability in the Lycopodium obscurum complex in North America and Eastern Asia. And when he graduated, you might not have known who he was. There he is in the middle of that picture. Looks like the wild man from Borneo there. But that's Jim. And I have to tell a personal story now. When Jim was getting ready to finish up at Miami, I was a freshman botany major. And I went to a botany picnic. And it was an interesting time in the early 70s. And at the botany picnic at one point, after we'd all eaten, there was a plate of brownies that was passed around. And I took one, and I took a bite. And this unknown individual leaned over and whispered in my ear, "You might not want to eat that." So, you know, I suddenly, it's like, why? I'm a Midwestern kid. I don't have any concept of why I should eat this brownie. But I didn't. And then later on, when the little silver tray came out with little white cigarettes on it, he leaned over to me again and said, "Would you like me to take you home?" And he did. So Jim Hickey saved me that day. So thank you, Jim. But that was my first introduction to Jim Hickey. All right. So after Jim left Miami, he worked at Harvard in the herbarium there for several years. And there were people there who were really influential again in his life. Alice and Rolla Tryon, Norton Miller, and I never say this name right, Xu-Ying Hu, who is a professor there, were very influential people in Jim's life. But there were also other individuals who were there. And some of the people who were hanging around at that institution at that time, Peter Stevens, Mike Donahue, Cal Sperling, Mike Froelich, Mike Campbell, Walt Judd, Dave Barrington, and so on, were some of the people that were interacting with Jim in his daily life at that institution. So there's a picture of them being very studious at some point. Now, Jim did tell me a story about this, and I probably won't get it right, but you know, when you're an enthusiastic young botanist and you go to a bar, what do you talk about? Well, in this case, they were at a bar, they got really rowdy and eventually got kicked out of the bar because they were so noisy, because they were arguing about the steeler types of some sort of fossil plant. You know? I mean, that's the kind of thing we all got kicked out of bars for, right? So I probably didn't get the story right, but it was something like that. So that's an important phase in Jim's development. There's Dave Barrington and Hugh Churchill and Jim Hickey from that era. All right, eventually then Jim ended up in a PhD program at the University of Connecticut, where he worked with Greg Anderson, who was known to Hardy Eschball because they had the same academic advisor for their PhD. And so Jim ended up working with him. And his dissertation was on revisionary studies of neotropical isoetes. So here's a plate out of that, one of the publications that resulted from that, just showing different spore morphologies in this group. So Jim learned a lot during this time, including computational skills, which really stood him in good stead. And he honed other skills. Darts. When I first met Jim, he used to take people up to the Circle Bar in the alley uptown and play darts. And so this just really struck home when I saw this picture. And there's a whole article about it that goes along with this picture. If you want to see it, I'll share it with you sometime. So eventually, then, Jim ended up at Miami University, and he was a visiting professor there. And I have to admit that I took Jim's job from him. When he first applied to work at Miami, he'd applied for the collections manager, essentially, job at Miami University. I also had applied for that position, and because I was already internal at the university, I got the job. And Jim keeps reminding me of that from time to time, that I stole his job. And he tells me I stole other things from him too, but I don't know what that means. But Jim worked his way through the system, got tenure, became the graduate advisor, and really worked hard to assist and get these graduate students through the program at Miami University, and had a really big impact on the lives of all these students that he touched, and many of you are in the room today. He did then finish up his career as chair of the botany department right before we were merged with the zoology department to become biology. Now, Jim, over the course of his career, has done field research in many, many different places, and this is just a list of some of the countries that he visited. And there's a picture of Jim with Susan Spratt wading through shoulder-high Pteridium, Jim down at the bottom looking very intently at some sort of flower. I don't know what it was, but he did a lot of traveling, and this impacted his research dramatically because he collected materials in all these places, described organisms from some of these trips, and so on. So I said I'd get back to snakes. Well, if any of you have ever done field work with Jim, you will know that one of the things he does is turn over logs, turn over boards, turn over rocks, turn over pieces of sheet metal, turn over cardboard, whatever is there, because he wants to find snakes. And he does, he finds snakes. Here's a picture of Jim in Belize when we taught down there holding a coral snake. And I remember one time when I was really, really sick because I'd been bit by a whole bunch of ticks and I was stuck back at the field station. He and the students went out and they collected a bunch of plants and brought them back for me to identify. And I was going through, sitting there really sick, telling him what all the names were, coral snake, so on. He had put a dead coral snake in the bag, thinking it would faze me, but it didn't faze me at all. And then here's a picture of Jim in the Bahamas with the brown racer, which is a toxic snake, but the fangs it's a rear fang, so you'd have to stick your finger down and start to get bit. But I do have a picture of Jim getting bit by a brown racer. Danette, his wife, is in the background looking horrified as Jim is being bit, but they're not toxic unless you really want them to kill you. So he was okay. But this is just Jim. And I asked him one time, why weren't you a herpetologist? Because you're so interested in snakes. And he told me this story. He went with someone to Florida, and there was a place called Reptile World or something like that down there. And he saw this big cottonmouth on a stem surrounded by this body of water that was supposed to keep it in place. And he said that big old snake looked him in the eye, and he had a come to Jesus moment. He knew if he studied snakes, he would die. And then later that night, when he was pushing a tent peg in with his bare foot, a coral snake crawled under it and just sort of cemented that in place. So he wasn't a reptile guy after that. although he still loves them. Right, Jim?
Yep.
All right, so Jim has published a lot of papers. There are 55 that I could locate, 55 papers, reports, and so on, presented at least 40 presentations at various meetings and so on. His students, along with him, have been awarded the AFS Wherry Award 3 times. He's gotten grants from all over, USDA, NSF, and so on, and served as editor for the American Fern Journal and Terminologia at various points in his career. And so there's been a lot of scientific output. He's also named a lot of species in his career, and here's a list of isoetes that are new species or new combinations. And if you're wondering what that person is doing on the left there, that's Jim in 1980 on a field trip, I think with Hardy Eschbaugh, out in a high elevation wetland in his underwear collecting isoetes. Fortunately, it's a discrete photo. He's also worked with students on other genera and other plant families, flowering plants even though they're gaudy. Adiantopsis, Adiantum, this Lycopodium from his master's degree, the genus Pleopeltis, and so on. So done a lot of work in various fern genera. And so it's been great. So here he's also got things that are named for him. Epinomi, I love that word. Adiantopsis hickey, published by Melanie Link-Perez and colleagues. Dendrolycopodium hickey. Isoetes X hickey, which of course is a sterile hybrid, but I would never say that to Jim, it was sterile. Polybatria hickey, and then there's a Trifolium hickey, which will be published this fall, and Jim doesn't know it, but there's going to be a Thalipteris X hickey that Melanie and I are going to be publishing soon as well. It's a hybrid from the Bahamas. Jim really loves teaching. And this to him, from my talking with him, is probably what he considers the most important aspect of his career. The influence that he's had on undergraduates, graduate students, and even those of us who were faculty, because we did take classes from him, and even though he gave me a B, I still like him. All right, so he taught at Bridgewater State College a couple years after he graduated. He taught at the University of Connecticut, of course, as a graduate student. He taught at Miami University, both branch campuses, and the Oxford main campus. He taught at least 30 different courses that I could figure out while he was at Miami University, and he taught all levels of peoples over the course of his career. And I'm sure that some of you who are in this room who had him as an undergrad could say what kind of an impact he had on you as an individual. He said he taught non-majors, majors, faculty. He taught lectures in laboratories and field classes in Belize and the Bahamas. I have to say he once taught a cladistics class that several of the faculty took, and I think the faculty were about ready to kill him by the time they were done because it was so hard. But he has standards. Jim also did undergraduate research projects with a lot of people in the department, including some of the people in this room who did undergraduate research with him, and that really started them out on their scientific careers. He also received teaching awards, and a couple that I listed here, the Star Award from the Student Affairs Office at Miami's campus, and the Distinguished Educator Award in the College of Arts and Science at Miami University. And his talk that he gave when he accepted that award was really a really impactful speech that he gave. Although I don't know that the administration really heard it. But one of the things that to me had the most impact in our time teaching together was this Flora of the Bahamas that we taught for over 20 years. We inherited this class from TK and Hardy. And so we were the second generation, and now I am training a new generation of faculty to take it over. So there's going into a third generation. It's been taught now for 42 years, I think. So it's been going a long time. But this particular experience for me was the most impactful in my time at Miami University because of the way we taught the class, the way the students learned and interacted with us and with their environment that they were in. It was very important. And it also led to, for us, a lot of research projects, and many, many, many research papers and projects have come out of this. And Jim and I were working on a Flora of the Bahamas that we still at least hope to keep going on. Jim had a lot of students over the years, 10 master's students, 10 PhD students, and there are pictures of most of them there. Patrick Garrett is one who's current, who's working with Rich Moore as well. Yeah, you can see some familiar faces in that suite of pictures. Jim also has a lot of friends and colleagues who really appreciate him. And here's a picture from Jim's retirement party. There's a picture with T.K. Wilson looking rather stern, I think. And a picture of me with Jim helping some people move there. So Jim's impacted a lot of us at Miami University. So I'm going to include here his family. And this is a group of people who've had a tremendous impact on him. And if any of you students ever went to lunch with us in the 90s, you know how much impact our children had on us and how much we talked about that and how some of you said that was the best form of birth control that you ever saw because we talked about them. But Jim's kids who were there, he didn't mean you. So Alex, Danette, his wife, Sean, Alex, Ryan, his sons, and his grandson, Wyatt, all really are, we really appreciate their input, and they've made Jim the strong man that he is today. So I'm going to close with this quote that I started out with early on. Two qualities that often blend: a good sport and a loyal friend. Thank you, Jim.
Thank you, Michael.
Well, that was awesome. Thank you, Mike. We're gonna keep going with the rest of the talks this morning. So up next, we're gonna bring up Ethan Freed, who's next in the schedule. This talk on learning Flora. Let me just figure out how to start the show. There we go. Learning Floras, building gardens, implementing plant conservation. Legacy of Jim Hickey at Miami University. With that, I'll let Ethan take it over.
All right, thank you. I want to start out by asking how many here actually did go to the Bahamas with any? All right, so you're going to see a lot of what came of that. Jim Hickey was, for me, a good friend. He was a great person to argue with. I know some of you guys saw that in him as well. But for me, to be able to show you this is because he was such a good friend. For me, Jim was the person that would always sort of walk me back from the edge. I would never have finished without Jim. So I was there in the 90s. You've seen this picture already. For those who've been to the Bahamas, you were there with these people. I worked very intensely with Mike and Jim. I was their first TA when they taught the course and taught with them for 5 years. So, I knew Jim in the lab, but I mostly knew Jim more in the field and to argue with and look at snakes and things like that. He taught me a lot about learning plants, about how to figure things out, which subsequently led to a lot of the stuff that I'm going to show you. All right, so most of you are probably familiar with Andros. You were there at 4FAR, you took the course. I did all those things and really just fell in love with the Bahamas and had the opportunity through the university, and particularly the herbarium, to continue to get funds to go to not just Andros, but to most of the islands that are in the archipelago, collecting for herbarium specimens, and then now for the projects that I'm currently on. I love the islands, I love getting around, I love the plants there, and it was just became what I did. So I end up here on Eleuthera. It's on the sort of northeastern part of the Bahamas. I've been living there now for 10 years on this project. So what did I end up doing? So this is Shelby White. She and her husband, Leon Levy, have been coming to the island of Eleuthera since the 70s. Fell in love with it, bought a house there in the 80s. Unfortunately, Leon passed away in 2003, but he left behind a very large legacy, a very large foundation. He was involved with Wall Street. And it's a philanthropic foundation meant to be entirely given away. And one of the things that Leon had always been fascinated about was bush medicine. They were going to the Bahamas in the 70s and stuff like that. It was very prevalent then and was very fascinated by it. He and Shelby are also very big supporters of botanical gardens, in particular the New York Botanical Garden and the Brooklyn Botanical Garden over the years. So after his passing, she decided that she wanted to build a botanical garden in the Bahamas on Eleuthera. And she got in contact with the gentleman on the right, that's Eric Carey. He's now the executive director of the Bahamas Natural Trust. Good friend of mine, I'd known him since the 90s when I was traveling around. And he called me up and said, Ethan, there's this lady. She has a lot of money. She wants to build a garden. You should come meet her. So I did. I met her in 2006. She spun me this tale of wanting to build a botanical garden that was focused on Bahamian native plants, bush medicine, and early childhood education. And could I go look for a great piece of land that you could build a garden on Eleuthera? So I went out and surveyed and found a really nice piece of land, great diversity on it, lots of endemic plants, orchids, bromeliads, things like that. So we ended up building the Leon Levy Native Plant Preserve. And I'm just going to go ahead and start talking about what we did. So we started with 25 acres. We just recently purchased another 5 to bring it up to 30. We started working on it in 2009, clearing land, doing construction and things like that, and then opened our doors to the public in 2011. At this point in time, I have over 450 species of plants in our living collection, and about 1/3 of the total flora. So what did we do? We built trails. Most of our site is actually intact coppice, so only about 1.5 acres of the 25 acres needed to be worked on. So we put in 1.5 miles of trails throughout the site. We put in a mangrove boardwalk. You can see all 4 species of mangroves that occur in the Bahamas. You can walk students right up to those mangroves, really show them all the characteristics that are there. We put in medicinal plant displays, and this work was actually based on the work from another Miami University graduate, Laurel Ritchie, who did her work on Cat Island and Long Island. We were able to use her dissertation and go through and develop these beds. We did a lot of display beds. If you want to showcase hundreds of plants, you need places to put them that are not just in your forest. So we've been putting in a series of beds, primarily focusing on endemics, but largely it's also ones that I find, if I can collect them, I will. If I like them, I will as well. The goal is eventually to have 50% of the flora in our collection, to have all of the endemic species. I'd like to have at least one collection of every family in the Bahamas. I don't know if I will get to that. We did a lot of restoration. So in the beginning, a lot of area in here was filled with invasives. And when I got there, I said, cut them all out, which left a lot of space open. But over a number of year period, we brought in over 5,000 plants, but we brought in 3,500 and been growing our own since then, and replanted it and turned it back into coppice. So it's done in a way that not just mimics, but recreates a forest system. So you can look at areas like this. This is our parking lot area, starting in 2010, totally cleared out to what it looks like today. We put an education pavilion. She had a very strong emphasis on early childhood education, so a lot of our focus is to bring students in, have them in a location where you can go out and you can look at things and then come back, talk about it. It's also a pretty great place for a party. We throw a pretty rockin' Halloween party for kids and a Christmas party, and we do a wine and art affair as well. We put in a welcome center with a gift shop and office space and things like that. We put in a tower so you can climb up. It gets you up above the vegetation. You look out across the island. You're actually in the top of the canopy to go check out things. We inherited an old cistern and decided to turn it into that, which is great because it's fresh water. It's true fresh water, so I was able to travel around the country, collect freshwater species. I had to build a new system for freshwater plants because we have these awesome turtles in the pond, but they eat some of our plants, so I had to build a second one that was turtle-free. We put in an edible history section. We wanted to talk, even though almost none of these are native to the Bahamas, there's a long history of farming in the Bahamas, so we wanted to build something to showcase not just what people were growing, but talk about how they got to the Bahamas, what was likely there with the Lucayans, what was brought in with the Spanish, what was brought in with the British, what came in with the African slave trade. Put in a weather station, doing 7 different variables, collecting them in 5-minute, 15-minute, 60-minute increments, as well as 24-hour increments. This was one that we decided to do because we thought we should really be recording all the different climate that's out there. I went with 5-minute and 15-minute increments, as well as 60-minute increments, because it allows you to get at storm intensity. One of the things that we know climate is changing, we don't necessarily know how climate is changing, so we want to record at a finer level. I was going to do 1-minute data, and the tech that set it up looked at me and thought I was crazy. He's like, this is a ridiculous amount of data. To tell you the truth, it really wasn't. After 6 years of continuous operation, we're only up to about 150 megabytes of data, because it's really all just ones and zeros. I'm thinking going back and doing the 1 minute. All right, so I want to quickly talk about our science and conservation program. So I was there, I did the surveys for the land, I was there during the construction and oversight and stuff like that, and then opened our doors to the public, and Shelby was like, Ethan, what are you gonna do now? You have to do something. I was like, well, I was done, I was gonna leave. And she said, no, you can't leave. Develop a science and conservation program. So had to sit down and go, well, what do you do in science and conservation? So in terms of, for a botanical garden, I came across this thing called the Global Strategy for Plant Conservation. Are you guys familiar with the Global Strategy? Some of you? All right. This was one of those places that you would think I had known about this when we built the botanical garden, because our site is so well situated to implement the Global Strategy for the Bahamas. So the Global Strategy, for those that don't know, it's part of the Convention on Biological Diversity. It's structured to give countries a list of targets to try and implement. It's done in a way that when you hit those targets, you have them fully done. For the Levy Preserve, we are directly focused on 9 of those, and the National Trust on another 4. The rest are done or not done at the governmental level, should we say, and some of them just don't apply to the Bahamas. I'm only going to talk about a few of these. I cannot go over all of them, but the ones that I found to be the most important. So target 7 is about ex situ collection. So we know there are areas that we cannot establish parks to protect these species, so to have them in, not just in your collection as, hey, I've got this plant, but actually have them in cultivation. So we built a lath house. We don't need greenhouses in the Bahamas, and I've actively focused on going after the endemics for this program. We have 89 species of endemic plants in the Bahamas. 31 are known to just a single island, and then there are 19 to just 2 islands. Those single island, double island, or endemics are our biggest target. Those are the ones that are most threatened. This is a good example. Sapirus corellii. This is really a single location. It's a dune species, so the habitat's only about 50 feet wide, and it's about a mile section of shoreline, and it's all in privately held lands, and it is being rapidly developed. This one will likely lose in the wild within this decade. And there's at least 6 here that we know we will never be able to put into parks because they are all on privately owned land. We'll never be able to put parks around them. So what do? I do whole plant collections. Since nothing is leaving the Bahamas, CITES doesn't apply to me, so I can take what I want for cacti and orchids and stuff like that. So I will, if I can, collect the entire thing. Others, I will do seed collections, and this is really what I try and focus on, is seed collections, trying to get a higher level of genetic diversity in our plants. And I could never grow orchids from seeds, that's for sure. So what do we have now? Of the 89 species, we have 46 in the collection, our living collection, so about half, and 14 that are in cultivation, where we actually have produced a large number of them beyond just having them in our collection. Success stories. Of those single island endemics, I now have 6 of them in our collection and 5 in cultivation, and another 5 double island endemics. So these are them. I'm going to speed up a little. So we moved things out of our lath house. We now have a nursery area. We have now 75 different species in this collection, and we started doing things with them. I started what I call a forest on a pallet project. This is one where we can palletize plants. They're in 3-gallon pots. I can get them off Eleuthera on a Thursday. I get them to the capital on a Friday. I get to another island within 24 hours after that. One of our big projects here was the National Art Gallery of the Bahamas. They had a very heavily destroyed piece of land right in the middle of downtown Nassau, the capital. They wanted to put it in a sculpture garden. They were a bunch of art people. They had no idea what they were doing. People gave them all these big estimates, hundreds of thousands of dollars. I met them and said, no, we can do this much easier and much cheaper, and started shipping them over. And this is what it looks like today. So it's all cleaned up and it's growing into a forest. This will be a fully forested probably in the next 5 to 10 years. It already brings in a lot of wildlife right into the center of downtown. Target 15, this is a big one for me. Training people, having capacity in the country. I got to do a lot of stuff in the Bahamas because I knew my plants, and there wasn't a Bahamian that could do that. So we got funding to do an internship. So every year we run what's now a 9-week natural history internship. It's only for Bahamians that are enrolled in college in the sciences. We've now had 17 students through the program. I want to take a little credit for Mark Daniels, another Miami University graduate. He wasn't part of our program, but he was certainly part of our training. We do Caribbean dry forest ecology on the site. So I got funding to set up permanent forest plots. We now have 3 20-meter by 20-meter plots. All the trees are tagged, identified, and then we resurvey them every year. This is what our data is starting to look like. We had essentially a forest system that was close to climax. So we're not really getting a lot of change over time. This is what our 3 plots, and we talk about our dominant species. So even though we have 3 plots and they're not all that distant from each other, less than 200 meters apart, they're actually quite different in their dominant species. All right, so what kind of continuing projects am I working on? Certainly continuing the global strategy, the forest plots, living collections, starting to look at development of a Flora of Eleuthera. Hopefully Mike will be down visiting, working on it. We're going to start doing seed banking as we get our new facilities up in order, and we're hoping to rescue the Bahamas National Herbarium this year. It's been in terrible disrepute, I guess we'd say. We have space for it now, a new building. We have support from the foundation, so as soon as we can get government to agree to let us have it, we will take that over and reestablish it. We also are, of course, looking at long-term climate data. We will continue looking at short-term stuff and collecting our forest plot data. We do a fair amount of entomology on the site as well as ornithology. I'm not involved with that other than getting the entomologists and ornithologists out there. I'll just end on that note. Before I finish, I want to let you guys know that we have a natural history, a terrestrial natural history of the Bahamas book coming out this fall. It's going to come out October 15th. It's from Cornell University Press. Those of you who still do teaching or anything like that in the Bahamas, it's going to be a great field guide for anybody. So, thank you.
Good job, Ethan.
I encourage if any of you guys get a chance to come to the Bahamas and Eleuthera, come visit us there. So, a couple questions.
I was wondering, of those students who did the internship, have any come back and, you know, been full-time to work for you there?
No, they've gone actually on and worked at other positions within the National Trust. Yes.
Are there accommodations for students, like from the United States, to come and stay? You brought a student group?
Not yet. So we have that for part of our phase 3 is to build a dorm that should accommodate up to 24. Like to think that that's up and running in 2021. So it'll be that. We'll have research facilities and things like that as well.
Thank you. All right, so we don't have the last, just FYI for everyone, the speakers, so we've got a little, we're being a little elastic with the times and the schedules because the last speaker isn't here today. And so trying to give everyone the full 15 minutes with transition. How's that? All right. Is this you?
That is me.
All right. So up next we have Steve Rybczynski.
Ribs is fine.
There we go.
But you got it on the notes.
And he's going to tell us about planting the seeds. Oh, I don't know what that means. Okay. Planting the seeds of discovery. I'll let Stephen take it away.
Thank you very much. Well, hi. I'm really happy to be here. I do feel a little bit like Frodo at the table of Elrond, but we're going to make do. I want to highlight one of the aspects about Jim that I felt was most important for me, and that is his passion for inquiry. I had to write this down because I didn't want to forget the details of it. His passion for inquiry, as I remember from those lunches, is only rivaled by his passion for complaining about anti-intellectualism. And really, I want to talk about this unquenchable curiosity. Inquiry is a word that has a lot of baggage. I'm going to try to talk a little bit about that, and I'm going to do that by showing you how the most recent national science standards see this. And so this is going to be a description of a lesson using this particular system to address these national science standards. NGSS has been out for a few years now, K-12 teachers are still struggling with implementation. And so these kinds of lessons where a person has time to really develop something are really valuable. The newest standards are not the laundry list of teaching standards you may have been familiar with. You were able to cover something about the process of science by using a system that was not necessarily realistic, like what's the best laundry detergent? And you can do a scientific study about that, but this is a little bit closer to, I think, what they had in mind. There's three dimensions to the next gen science standards, and parts of this are pretty familiar probably. These core ideas, this is the content that we're all familiar with. These science and engineering practices are the things that scientists do. Things like communicating information, using mathematics, using models. And at the end of all of this is the ideas that link discrete science disciplines. What does physical science have in common with life science? Things like energy, things like scale, patterns. And so these standards pull all of these three dimensions together. And so how do you do this? You've got 36 kids, 7 hours a day. What do you do in the classroom? Well, where do I fit in? Right, we'll get to Redbud here in just a minute. So here is the actual standard. This is the performance expectation that they want high school students to be able to do. Use math to represent explanations of factors that affect carrying capacity of ecosystems at different scales. There are a whole bunch of these. Now, there are specific cross-cutting concepts, disciplinary core ideas, and science and engineering practices that align with this particular standard. So this one asks us to use math and computational thinking. It wants us to talk about the interdependent relationships in ecosystems. This is where the plants come in, because the cold reality is that plants don't factor in very much in the next gen standards. They stop talking about flowers during 5th grade. They talk about some stuff during middle school. At the high school level, all they talk about is photosynthesis from a chemical perspective. And so it's up to individual educators to take these stories and incorporate them into this. Scale, proportion, and quantity. And so here, we need a phenomenon. We need something to talk about all of these things about, and Redbud is going to be our model system for this. How you doing, Jim?
Doing great. How are
Good.
you doing?
I'm sweating. It's okay.
Take the damn coat off.
Take the coat off.
No, I don't want to. I don't want to show up. If you really knew me. This lesson is going to be in what we call the 5E learning cycle. This is a cycle that Jim first introduced me to. It involves engaging somebody with a phenomenon, having them explore it, then explain what is actually happening, and extend it then to novel situations. Evaluation occurs throughout. So the way that I envision this is that you take students to an actual tree. Let's take a look at one. And I ask them, what do you see? How many people have ever had Jim say something along the lines of, what do you see? What are you observing? What's your question, right? And so what questions do we have about this? And I try to shepherd the students to, well, first of all, that's an awful lot of flowers, right? It's a very pretty tree. It's better in person, but the picture works pretty well. And observations lead to questions. And so some of the questions that students tend to bring up are, why in the world does this organism produce so many offspring? This is a redbud that was in my mother's front yard. These are not leaves from the previous year. These are fruits. The fecundity of this thing, in my mind at least, is amazing. But this is a tough question to answer. It's a causal question, and especially in a situation where you're teaching somebody in a semester how to answer these types of questions, this is really hard to get to. So I try to get them to break it into more of a descriptive question. And if you can answer some descriptive questions, you may be able to bring to bear on these larger causal questions. So we come up with a plan for, well, let's make an estimate of how many offspring this plant can actually produce. What is this organism's reproductive potential? This is the most important part of this lesson, to actually let students explore the system. I have figured out that if you give groups of about 3 to 4 students about a half a meter of branch that's covered in fruits, that'll usually get you between 75 to 125 fruits. And this is a reasonable number to be cut open and the seeds enumerated. I like to use petri dishes because these seeds run like mercury. But students will immediately start to notice things. It takes about 30 seconds for somebody to say, oh, my seed's got a hole in it. And it doesn't take too much prodding to say that, well, what is this? What's going on? What is your possible explanation for this? Well, a bug went in. How do you know it went in? We don't. But let's just go with what our question is. How many offspring does this produce? This is clearly not going to grow into another organism. So we have to accommodate or account for these infested and aborted seeds. If you allow students to play with the system, if you leave the seeds and the fruits in a jar, things are going to start crawling out of them, especially if you collect them in the fall. I don't know if this is going to work. I'm going to run the video just in case, but I did manage to catch a bruchid climbing out of one of the seeds. It takes a surprising amount of effort, doesn't it? But it's going to pop out of there. And, oh, I can't do that, can I? I totally blew it. We're just going to keep rolling. So the beetles come out of these seeds, and the question is, well, what is going on here, right? And either you can tell the students, or you can, if you have the time, have them investigate about these individuals or these organisms, right? And so here are the beetles that come out. They're relatively small. That scale bar is 1 millimeter. The life history goes is that the beetles will deposit on these developing fruits during the summertime. I have a couple of red buds in my yard now. I found that dark, dreary, rainy, miserable mornings like the kind that we had today are the days that you're actually going to see these adults ovipositing on the fruits. I've only seen it a couple of times, but the larva hatches from the fruit, or the seed, burrows its way into the ovule, and then sets up shop and eats it from the inside out. The students use the data that they have collected to generate a figure. Here are some numbers with just gargantuan error bars, but it clearly shows that the number of infested fruit is almost as big as the number of viable fruits or seeds. And here now you can see how we started to address, we have used math and computational representations to support explanations of factors that affect carrying capacity at different scales. Now, this is just one standard. I've managed to hit 3 standards. I just don't have the time to go into each one individually, but I'm going to continue with this because other things will crawl out of these fruits. This is the individual. I tried really hard to catch it while it was actually emerging, but it's tough to use an iPhone through the ocular of a dissecting microscope. It worked out all right. I've managed to find at least 4, I think that I have 5 different species of hymenopterans that have emerged from these things. They are parasitoids, and in this system, they act as top-level consumers or secondary consumers. We've borrowed a system from the biological science curriculum study on food chains. We get students to see that there is a decrease in biomass as we move up. This does address one of the standards. And then this is the most fun part of this is I have them make this figure. I give them these words, the red bud, the bruchids, the wasps, decomposers in the atmosphere on Post-it notes, and I put them on a whiteboard and they have to connect the arrows. You would have no idea how few students actually recognize that it should be a cycle, right? All of the things should, but they get there, right? To wrap all of this up, I want to encourage everybody to show and then tell. Let's take a look at the phenomenon first and then come up with the explanation. It's these phenomena that drive the inquiry. If you have nothing to explain, what do you got, right? You have to start with that phenomenon. And I want to end with a Jim quote, that any well-built ship can stand a little rocking. I don't know if this is exactly right, but I know that Jim has a reputation as a curmudgeon and rather abrasive and this and that, and we joke about being nurturing, right? But I can honestly say that I would not have survived my first year teaching at Oklahoma without Jim's support, the late night phone calls, and everything. I'm going to leave it with a well-built lesson can also stand a little rocking, and if you don't have a red bud, there is no shortage of other plants that you can investigate. Thanks to these guys who paid for my trip.
Questions? Is there any between the transitions? All right.
One of my fingertips.
There we go. It's up. You're up next, Julia. It is the ferns
My goodness.
on the schedule.
So,
No, it doesn't advance.
Julie?
Pointer.
so is it-
Jim said hi, Julie.
Hi, Dr. Hickey.
All right.
I'm sorry, I cannot really call you Jim. And Dr. Ashma, hi.
For those of you that just are going in and out, we're just sort of being a little elastic at the time because we have some opening in the schedule, but mostly sticking at about 15-minute talks, so we can keep everything close on time. And we've got Jim on the video, so for those of you that are asking questions, he's judging you.
Oh, no.
All right. Well, with that, I'd like to introduce our next speaker, Julie Barcelona, who's going to tell us about fern and lichen diversity in a fragmented forest. Thank you.
Okay, good afternoon. Don't worry, I'm still working on ferns, although I'm working on the grassroots level. You know, there are a lot of NGO work in the Philippines right now because a lot of surveys have not been done. The Philippines is one of the most degraded countries in the world in terms of forests. We have less than 10% of our habitats, but a lot of these have not been explored botanically, especially in this island where I worked as a consultant with a USAID funded project of an NGO, the Soil and Water Conservation Foundation. The executive director is actually an American. He used to be a Peace Corps volunteer and then married a Filipino and stayed there and established this NGO. So I'm going to talk about the fern and lycophyte diversity in a fragmented forest. This is one of the most comprehensive checklists that was produced for this protected landscape, and also the conservation recommendation for this protected area. My co-authors are two young research assistants of this NGO, and of course, my partner in progress, Mr. Pelser. I've seen this photograph used in several fern talks, and I'm really glad because I actually took this photo. So this is Chilia ferox that I took a couple of years ago. So the protected area, this
One advancement points.
oh, can I? Okay, good. So the island of Cebu is located in the central Visayas, and it is one of the, if not the most degraded island in the Philippines. And I also put Bohol there because in the early 2000s, right after I graduated, I did also a survey for the same NGO of the another protected area there, the Rahasikatuna protected landscape. And I actually published the result, the American Fur Journal or something. And the protected area is located in the central part of the island. And it covers about 28,312 hectares. And if you notice, this protected area is inside all of these cities. So there's not a lot of forest left. See that? And so I wanted, you know, the NGO wanted to see if there's still any fern at all or any fern and lycophytes present that would help them convince the local government and the Department of Environment and Natural Resources to sort of at least save these patches of forest. And before it became an encompassing Central Cebu protected landscape, there were several protected areas within it, and then they made a one big protected area. And if you notice again, there are 61 barangays. A barangay is considered as a barrio, or the smallest political unit in the Philippines, in the different cities. And historical plant collections, I looked at my database of Philippine ferns and lycophytes that were collected in Cebu, and there were only about 111 collected specimens, so very, very meagerly collected. And one of the very first botanists who went there and collect was coming in 1841, and then Maduli et al., as part of the Flora of the Philippines project, also went there in 1997. So there's not a lot of collection, and a lot of them also were not really pteridophyte focused, but rather vascular plants. So these are the young research assistants of the NGO, and this kid is like, it's like a toad. He doesn't listen. He doesn't have any gear at all, and he would just go in there. So between November 2016 and February 2017, we did intermittent field work, and we went to the different sites. This protected area is mainly limestone. So we had to go in these very, very sharp, almost dog-edged substrate. So these are the sampling areas, and these are the places where we went. And I was really lucky enough to bring somebody who can actually take photographs, good photos of the ferns. And in summary, we were able to collect about 151 species in 6 varieties in 61 genera, and 24 families. So this increases the current knowledge of the ferns and lycophyte diversity in these protected areas threefold. And what we have observed is, although the forest fragments sometimes are very, very small, a unique combination of species are present that you could not see, in other adjacent parts of the forest fragment. So that's very interesting. And again, many species are confined to a single forest and then are only represented by very, very few individuals, or if not, one plant. So this is just a checklist of what we found. And previous studies, I think run by some consultants, included these within the protected area. These are common species. I can go to many forests in the Philippines and stumble upon these things, and I've never saw them. I don't know, I've never seen any specimens. Some of these are actually also found, like across the pomarium. This is a back mangrove species, so I don't expect it to be in the forest, in the limestone forest. So anyway, so what I did was, it's very difficult to evaluate something without comparing it with something, of similar area. So when I compared the species diversity between the Raja Sikaduna protected landscape and the Central Cebu protected landscape, they have, more or less, I added the same number of new species records there. And in contrast to the CCPL, RSPL is a huge intact protected area. It's not fragmented. It's a big contiguous forest. And in RSPL, it has many species that are locally abundant as opposed to the CCPL. And RSPL is almost 1/3 of the area. And the elevation, there's also elevation differences. And I have to admit that I spent more time in RSPL than in CCPL. So I'm just going to show you some of the photos of the ferns there. And when I compared the two protected areas, they share about 45% of the species that are common to both. So one of the most, noteworthy discoveries is a new record for the Philippines. This is a species of Asplenium that was previously reported for South China, Burma, Southeast Thailand, and Malaysia. And we've only seen this in one small patch of forest. And when I saw it, I said, I've never seen this. I have done a lot of explorations throughout the Philippines. And so, there's some species that would just knock you off those limestone, limestone forests. And this is Asplenium salignum. And before we went there, there was only one tree fern, Cyathea species, or Cyathiaceae, that was reported, and this was Phaeropteris glauca, which is one of the most common one now. Used to be Cyathea contaminans. And then we added 4 more: Sphaeropteris intigra, Sphaeropteris tripinata, and the Almarai. And I still have, it's very challenging, because in as much as I wanted to collect every single fern specimen that we encounter, we also are not currently based in the Philippines. I live in New Zealand, so we have been there for 10 years. And so I had to rely on really good photographs to later identify my specimens. And I do pretty well, except for these things that may even be, new record. But in this project, we did collect a lot, and the specimens are currently in one of the institutions in that island. So when I go back, we're gonna identify this. And I'm just gonna show you all the amazing ferns in that area. Beautiful. So this is what the protected area looks like. When they established this protected area, there are already communities within it. So it's not like, it's very difficult to manage this unless you involve the community. And, education is really very important. And then this is from another barangay. You see there's a lot of conversions going on. Very, very small forests are continuously being cleared for flowers. It's Teresi. So I don't know how this protected. And here, they've gotten rid of all the native limestone, mouth of the cave dwelling species and converted it into a backyard garden full of exotic plants. And some of them are actually invasive. And charcoal making is still very, very rampant there, and you cannot question them. That's, when we went to this barangay, it was one of the most difficult trips I've had. It's just very, very inaccessible, and so the people there don't have anything almost to rely on except to cut the forest and convert them into firewood and collect some of the animals and sell them, birds. And so these are the recommendations that I think, this NGO I heard that they were able to successfully convince the LGU, the local governments and units in the Department of Environment, and that, before we can do something or solve any problem, we should acknowledge that there is a problem. And there has to be an establishment of a strict protection zones for this remnant forest. And hopefully, through time, that can be extended, those primary habitats. And awareness, promotion of environmental education. I think that's really very important in the Philippines, because before, when I was a student there, I've never heard of, environmental education. We always heard here that the Philippines is full of natural resources and it's rich, I never heard the other side of it. So A lot of NGOs now are very active in promoting environmental education. And it would be nice to also do similar expeditions to other parts of Cebu, because there are some good forests, although small, outside of the protected area. And assist natural regeneration in severely degraded forests. So photographs obtained from these expedition have been uploaded to our website, the Coast Digital Flora of the Philippines, which currently we have photo documented, I think more than 50% of the fern species and about 42% of the vascular plant species. So that's what I have been doing, a lot of volunteering and grassroots surveys and education. Thank you.
Thank you, Julie. Julie?
Oh.
We have time for a question or two while we transition over to the next slide.
Yes.
Have you been able to do any sort of predictive analysis, like a species area curve or something, as you go from plot to plot, to try to estimate how many theoretically you should have in the area?
Those are the things that I wanted to do, but the problem is we're always limited with time, you know, and it's nice to do it when you know that you have enough time to actually, and I know that there were several people who did plots, you know, in previous studies, but then, you know, the result of the plots, there are only 4 species of birds in the entire. So I would like to do that, you know, to really see and objectively compare those areas.
All right. Well, thank you.
See you again.
She'll be back. She'll be back, folks. Don't worry. She'll be back for round 2. All right, and next up is Julie Ballinger telling us about rarity, carnivory, and promiscuity.
Oh.
All right.
It's
There
an
we go.
honor to be here with all my Miami friends and Miami mentors. And I have to tell Jim, the favorite class I took with him was plant taxonomy. And we drew all of our families, and I saved all my drawings. And now when I teach plant taxonomy, I make my students do all the drawings. And I showed them my pictures. So thank you, Jim. One other thing, he would always have extra credit. I would get 102 and 106, 109. I get 99 on my paper. Jim, why? He said, well, my grade book only goes to 2 digits. Not fair, Jim. I survived. All right, so I'm excited to talk to you about some of the work I've been doing on these rare insectivorous plants in the fall-line sandhills in west central Georgia. So fall-line, this is where Piedmont meets coastal plain. And you have this nice series of gentle waterfalls. So that's what the fall line. This is also the ancient coast in the southeast. So this is where we have massive sand dunes, some of them 400 feet deep. And as the ocean was waxing and waning over those times, you get layers of sand and layers of mud and layers of sand. And so you have these interesting hillside seeps, and all these interesting plants end up in this environment. As the ocean retreated, all these things were kind of left there, kind of stuck. So we have very unique combinations of different plant species in these fall-line sandhills. So our goal was to characterize these wetlands and determine if we had any new plant communities. And so we were comparing to what's on NatureServe and in the Natural Heritage Program for Georgia. We are documenting these species using traditional taxonomy. We also did some DNA barcoding, trying to build a barcode library of the fall-line sandhills area, both the uplands, the dry areas, and the wetlands. We're also looking at the hydrology of these sites and the soil types. Another goal was to develop some management recommendations. Historically, fire was very common on the landscape in the southeast, and that's been removed. So you have a lot of woody vegetation moving in. We've been working with Fort Benning, which is just south of Columbus, where I'm at, the Department of Defense, private landowners, Georgia Power, because as Georgia Power puts in power lines, they open these areas up, and then you get these incredible plant communities. The downside is the way they keep those areas open. They don't use fire, they use four-wheelers. They go in and tear up these sites. We've been working with Georgia Power and the Department of Defense to come up with some strategies for maintaining these sites and protecting these sites. So, okay, here's Georgia, West Central Georgia right here. This is the fall line sandhill that runs through the state, goes on up into the Carolinas and then back to Alabama and Mississippi. This is Fort Benning. This is where we started our project. And in the past, probably 10 years, Fort Benning has been purchasing thousands of acres. So this is 180-some thousand acres. They've been purchasing thousands of acres to the east in the sandhills. This is called the Army Compatible Use Buffer Zone. And so now we have access to all these new sites that were privately owned. And those landowners that were not interested in selling, Nature Conservancy has worked with them to put conservation easements on those sites to protect these plant communities. And the way we were selecting our sites, we were using aerial photography and looking for bog areas. We would go in and ground truth and then determine if we wanted to set up a site there. We had 11 sites. We followed the Carolina Vegetation Survey method. Nomenclature follows weekly. 2015, we did collect voucher specimens and we collected samples for DNA isolation and amplification. We collected soil samples looking at percent organic matter, pH, hydrology, the water content. We also estimated the landform index. So it's very different if you're standing on a hillside seep versus a beaver bog where you have a pool that's holding a lot of water. And then we were always estimating percent cover of water. This is what one of our typical plots would look like, a 20 by 50. And within that plot, you have these 10 modules. In modules 2, 3, 8, and 9, we had intensive plots within, and then there was nested subplots. So within those plots, every single stem was identified and then given a cover class. And then once we finished with those intensive plots, then we would walk the entire site and identify any other species we hadn't already recorded and give them cover classes. So we were looking at herbs, shrubs, trees, and then percent cover for each of those species. We analyzed our data looking using cluster analysis. We used PCR. Then we also use canonical correspondence analysis to determine those environmental influences on those species composition. I mentioned DNA. We have hybridization of 2 pitcher plants. One pitcher plant is common in our area. The other one was not previously reported, and we found this at several of our sites, the species at several of our sites. And then we also found hybrids. And then these are both graduate student projects. We had another graduate student building a barcode library of the fall line sandhill plants. The goal is to use this information. One of the reasons, I'm sure I shouldn't record this, but one of the reasons Fort Benning has been purchasing land is gopher tortoises are about an eyelash away from being listed. If they become listed, then Fort Benning, it will be unusable for training. They're going to have to move those gopher tortoises. And so understanding the preference, the diet preference of those gopher tortoises, if we have a library of the plants we find in the sandhill, we can look at gopher tortoise species and determine what it is they're eating and what they prefer, and then enhance those environments. So that's part of our goal. All right, so this is what one of our sites look like. I'm okay with water. I'm very comfortable in the water. What I'm not okay with is mud in water. One of my fears is getting stuck in this mud and drowning. So this really pushed my comfort zone. In addition, of course, we had cottonmouths at our site, and we had wasps. Wasps are actually worse than the cottonmouths. So when we looked at cluster analysis, our sites, this is based just on species composition, our sites clustered looking at wetland seeps, and then these deep, mucky mud sites, and then open water sites. When we looked at those environmental variables and applied that, it does look like we may have a new plant community. And it's most similar to what's called G1. This is your global ranking. So it's critically imperiled. This is the east, it's similar to the East Gulf Coastal Plain Muck Bog Association. That's not easy to say. And it does look like that percent water cover, the landform index, soil pH, and then organic matter. Those are the things that are really influencing the species composition. So the new bog, this new habitat is characterized by having high water cover. We do have some herbaceous indicator species, things like Sarracenia, Drosera, Xyris. If you know anyone who is an expert in Xyris, I hate it. It's a pain in the butt. And then there are these little woody vegetation, little islands of woody vegetation where we had Morella, Nyssa, and Lyonia. 230 different species, 110 genera, 69 families. 15 of these were rare to the state of Georgia, and 9 of these plants were carnivorous. And I'm going to talk about some of these different discoveries, but I want to point out the Sarracenia psittacina. This is the parrot pitcher plant. It's known in Georgia, but about 100 miles south of our site. So we extended the range significantly northward. And this is a pitcher plant that was hybridizing with our more common pitcher plant. We also found Ericolin texensis, which is one of the bog buttons, and it was formerly restricted to the outer coastal plain. So these are some of the major finds. And I'll talk a little bit more about those. All right, so the red milkweed, again, G is your global ranking, so these are not impaired globally. SH is state historical, so this was thought extirpated from the state. We found this milkweed at 3 different sites. Again, these are big plants. They're over 5 feet tall, huge pink blooms. But no, people in the right minds aren't going out to these sites, so that's why they haven't been recorded. I'm convinced of this. Northern white fringed orchid. We had 2 plants at one site. This is one of those global, this is one of the hillside seeps, and Georgia Power contractors drove their ATVs through our site, destroyed one of the 2 plants. They got stuck. You could see they got stuck and they tore it up. So this is one special concern, and it's really sad to see our one plant messed up. So this Carex exilis, coastal sedge, this was a new species for Georgia, never been recorded in the state. I wanted to mention the Atlantic white cedar. And you notice this is, remember, this is the fall line sandhills right here. This is our area, Fort Benning, and this is probably Fort Gordon up here. Those military installations inadvertently preserve extensive species, different habitats, animals, and plants. So it's really interesting to live that close to a military installation. Anyway, these Atlantic white cedars, most of these populations were owned privately. Working with Nature Conservancy, many of these are now under conservation easement, so they are protected. I wanted to mention the pinguicula. This is, again, we're looking at these aerial photos. We can see a nice site. We know it's there, but we can't get to it. We walked along the stream. We found a little tiny opening in the woody vegetation. We pushed our gear ahead of us, and we crawled to this little opening, and it opened up into this shallow, sandy, braided stream just covered with pinguicula. This is probably the best population in the Southeast. Some other, the other insectivorous plants, a couple of species of sundew, and several species of bladderworts at our sites. Mentioned the molecular work. These are both graduate students. We did confirm that we do have hybrids between the psittacina and the rubra, the 2 pitcher plants. And then again, the idea of building this library, this barcode library of the fall line sandhill flora. Back to the pitcher plants. Using RBCL and microsatellites, Christina Caldwell was able to determine that both haplotypes in the hybrids were present, but she was able to also see that the hybrids were preferentially back crossing with rubra. Rubra is the pitcher plant that's more common in our area. Again, psittacina is the one that we extended the range northward by about 100 miles. And again, this is what psittacina looks like. Typically in open water, it lays down in the water. Rubra is close to the water, not usually in it, stands up. And then this is what the hybrid looks like. This is one of the morphotypes of those hybrids. And you saw every range you can imagine in morphology. she had over 300 specimens of the parent plants and the hybrids. Scott Silvis was working on the DNA barcode library using RBCL. He only got 62% species resolution. This is probably because 44% of our genera are polytypic. So the goal is to add other markers in there and further resolve those species. So again, we want to do more genetic work. We are continuing the taxonomic work. Like I said, I've got everything but Zyrus. That's one I can't get. Michelle and I would like to add 2 or 3 additional sites similar to the East Gulf Coastal Plain muck bog, but that's easier said than done. It's not even easy to say. We again want to develop reference conditions for this new combination of plants. We would like to have a restoration site on one of the areas that is now owned by Department of Defense, put some raised walkways in so individuals can go in and look at the orchids and all the insectivorous plants. And then again, it's really important that we develop those management recommendations working with Department of Defense, Georgia Power, and private landowners. So Meade, West Vaco, Fort Benning, and Oklahoma State University provided funding for these projects. These folks are all or were at Fort Benning. Kevin Burgess is one of my colleagues at Oklahoma State. Christina and Scott were graduate students. And Brad Wilson is actually a veterinarian from Atlanta who just likes going out into bogs. Any questions?
Thank you, Julie.
Thank you.
Back in the back.
We got time for a couple questions
Yeah.
here before we move on.
So when Citrusina hybridizes with rubra, it looks like there's a risk of it losing a lot of characteristics for its specialized trapping methods.
Yep.
So I'm just wondering if you know anything about the hybrids being less successful than the parent species.
Right now, it doesn't look like the hybrids are less successful. They seem to be doing very well. And like I said, they're preferentially hybridizing back with rubra, which is more common in our area. But they really do have different strategies. And when first time I walked into this area that had all these hybrids, I'm like, what the hell am I looking at? There was everything you can imagine, every variation you can imagine. So I was surprised that they were preferentially hybridizing back with rubra. But that's a good question. More work needs to be done, yeah.
Just wondering, out of curiosity, if you happen to come across any lilies when you were out there.
Yes, I think so. Actually, I don't remember. Yeah.
Just striking orange turf cat lilies.
No.
Very similar habitats in North Carolina with the margin species would be where sandhills lily grows in North Carolina, right in this kind of
what time of year would they be flowering? Yeah, and we're out there, so we did not see any of those big lilies. Yeah.
There would be a range extension.
It's born
like 6 plants are at Fort Worth.
Okay. Okay.
So you're on the other side.
All right. Yeah.
All right. Well, thank you. Okay. So our final talk of the first half here before the coffee break is going to be Julie 2.0, who's going to tell us about, oh, did I open the wrong one? It's a little sensitive.
Anything about red buds, so.
Yeah, you don't want to do that. That could be the way to go here, give each other talks.
Oh,
She's going to about large flowers and big secrets in Rafflesia.
Yeah, well, now that I think about it, I think, Dr. Hickey, you did not clone yourself among your students, but rather you gave us some additional push to be what we want to be or what we enjoy doing. And this has nothing to do with the first that I studied before, but then I spent almost 2 decades in studying this majestic parasitic flowers, the Rafflesia. So today I'm going to share with you almost the culmination of our almost 2 decades of study of Philippine Rafflesia, because I think everybody's ready to move on to something else. So large flowers, big secrets, unraveling the biological mysteries of Philippine Rafflesia. One of the things that I have remembered that Dr. Hickey told us students is, if you have to apologize for a slide, don't show it. Don't worry, I will still not apologize, but I'll show it anyway. So I worked closely with two other Miami graduates, Dan Nikrent of the Southern Illinois University. He got, I think, his master's degree with Cardi Ashba. And of course, Peter Pelser, who became my life partner. He was Dr. Watson's postdoc, and we met at Miami. And then he dragged me to New Zealand. Okay, here you go. Here's Peter, celebrating his 10th year at University of Canterbury in Christchurch. His South Korean PhD student made this cake for him with matching fly. There you go. That was really good. And of course, look at Dan Nicker. It's 2008. And you know, during this trip, he said, I ponder if I still wanted to go to field work, or my role in this project would just to sit down and write grant proposals. But that didn't happen. He just came back from the Philippines with an NSF granted funded project for a 1-month expedition in Southern Philippines. So he's still going. I took this photo in 2007. This is Rafflesia schembergiana. This is the largest flower in the Philippines that was discovered in 1882 and was never seen again ever, despite the fact that, of course, it's large. And there were several expeditions done in the type locality to try to look for this, but nobody has ever seen this. And I have been lucky to be in expeditions with the mammalogists in the Philippines, led by the Field Museum. You know, those are my expedition buddies. And one of the technicians in that expedition told me, there's a big flower where I came from. I never saw it, but my wife told me that, you know, somebody put it on top of her head and moved, showed it around and tried to sell it, but then it's kind of smelly. So anyway, I did not believe him. I said, you know, from now on, I would only believe in a story that has photographs as proofs. So Rafflesia is a total parasite. It's an holoparasitic plant. It doesn't have any roots, stems, or leaves, and it doesn't need it because it gets its nutrients totally from the host plant. And the genus, this is not an updated number, especially the Philippines but then because every year, you know, there's new species reported, although some of them are quite very morphologically similar to each other, but very, very many are, several of them are actually good species. So in the Philippines, before 2002, there were only 2 names recognized. Now we have 13. And I was involved in naming and describing 7 of these newly discovered species. And yeah, it was really good. And this is Rafflesia leonardi. So as a holoparasite, you would only see that there is a Rafflesia in the host plant when it started to go out as a bud, and then it produces. It's like aliens. They stay inside their host undetected until they're ready to reproduce. So Rafflesia is exclusively parasitizing the host genus Tetrastigma. This is in the family Vitaceae. And they are huge lianas in the forests. And in my years of expedition in different, you know, forests in the Philippines, seeing different Rafflesia, I noticed that Tetrastigma is one of also the most pioneering species. They can be found in very disturbed areas and also in the forest interior. And I was wondering why, perhaps one of the reasons why Rafflesia species have come out, you know, in terms of they can be dormant, because I've been to forests where they have never been seen for almost 100 years. But then when they stop you know, going and putting tents every now and then Rafflesia came up. So, and that's, I think, one of the reasons why we have seen so many species, because of all this opening up of habitats. And the tetrastigma does very well also in these habitats. They started to parasitize perhaps, you know, on the root system. Look at these. You know, these are all senescent flowers and autumn flowers. And they can start from the roots up to the stem, aerial stems of the liana. So they first show up as balls, small balls, and these are the buds. You know, they look like cabbage when they're ready to open. But yeah, they can, you know, Rafflesia flower in the Philippines ranges from about 14, maybe even 9 centimeters across to 80 centimeters. So there's a huge range in size. They are, some of the species are very, very smelly, although some are quite indistinctly smelly. But despite the fact that we may not be able to smell all of them, their pollinators do, you know. So the known pollinators of these flowers are different species of flies. And if you look at this fly, the yellow liquidy thing on top is actually are the pollen grain, the liquidy pollen that it took out. So the anthers are underneath this disc. And conservation-wise, some of the species are only known from one population. This is, for example, Rafflesia manilana, and it has also never been discovered since its discovery in 1842 and was later discovered in 2010, I think. So, you know, they've been there, but not quite. So most of the Philippine forests are experiencing really bad, you know, big threats, from logging to charcoal making, from mining. You know, most of the southern parts of the Philippines are being mined like that. Amazing. And in the northern Luzon, farming of cabbages and carrots and all that stuff. So because Rafflesia, most of the species are found in low elevation, below 1,000 meters, they are very, very prone to habitat degradation. So you cannot conserve what you don't know. So today I'm going to talk about, thanks God, Rand, there's a miracle, Randy's not here, the taxonomy and distribution of Philippine Rafflesia. Very recently, we published a paper on its phylogeny and biogeography with Danny Krent and also Ben Van A, who's now in Puerto Rico. And I'm going to talk about host specificity. Is it really true that a specific species of Rafflesia is only parasitizing one species of tetrastigma? And reproductive biology, what are the seed dispersers? Are Rafflesia monoecious or are they dioecious? These are the questions that 20 years ago we didn't know really. When I started, you know, when I first described the third Rafflesia species, now we are on the 13th, right? In 2002, we don't know anything about these reproductive biology and host specificity data. And then conservation genetics. We looked at 2 species and look at gene flow between the populations and compare the 2. So this is again Danica, Peter and myself. So there are 13 species of Rafflesia in the Philippines. This map we made, these sizes are relative to each other. And if you notice, the southern Philippines taxa are larger than the northern counterparts, although there's one small species here. They're all endemic to the Philippines, and only one species is found in 2 islands, 2 adjacent islands. They are also, the species are also notably absent in bigger islands like Mindoro and Palawan, and even Cebu. Now, okay, I'm very brave to present this, but to tell you the truth, I did not do this analysis, but I did a lot of the lab work, a lot of of course, the collection of the species. We had to divide the roles in this project. And Peter and Ben Van A and Dan Rickrent were the ones who really cranked up this part of the project. And after so many years, we started collecting for this project in 2008, I think, and then it took more than 10 years because every year, there's almost another new species. So if you look at this, Rafflesia probably evolved in Borneo and spread to the Philippines via Panay. So this is actually the island where it came from. I grew up here. And then from Panay, it dispersed to Luzon, and from Luzon, it dispersed to Mindanao, to the Bicol Peninsula, and to Samar. And one species here in Panay, Speciosa, also dispersed to the adjacent Negros Island. And about 36 to 6 to 7 million years. This is much, much older than previous Rafflesia estimates. And there are very few dispersal events between islands, and that the poor inter-island dispersal as explanation for the high island endemicity. So this shows you that the Philippine Rafflesia is monophyletic, and that flower size is a very poor indication of affinity, whereas geographic location is very strong indication of affinity, and that there's a very strong biogeographic signal. So again, the Borneo ancestor went into Panay. So D and E is Panay and Negros. And then, from there, it went to the different parts of the Philippines. Here in Mindanao, these are the larger Rafflesia species. And in Luzon and in Negros, the green one. Host specificity. What are the tetrastigma hosts of Philippine Rafflesia? Do individual Rafflesia species have multiple hosts? We don't know these answers before we really spent a lot of time in the field. And do individual tetrastigma species host multiple Rafflesia species? Or is there evidence of co-speciation between the host and the parasites? And are hosts infected by more than one Rafflesia individuals? Or so we looked at this. When we are in the field, we just want to make sure that we collect specimens. We looked for other questions that we may answer later, for future projects. Because once you get the permit, it's not infinitely that you will have the permit. So what we also started doing is collecting the tissues for Rafflesia, as well as collecting tissues for the host plants, as well as collecting tissues of other tetrastigma species that are found in the area but are not infected. So, we collected all these things. And sometimes if there are many, many buds and flowers in one host, sometimes you can have 30, So we don't feel very bad to collect more than one sample, tissue sample, and we start from the roots and then sample some, from the ground level aerial, and then so on and so forth. So we did these in host plants that have a lot of buds and flowers. So this is what we found out in those sampling, that we encountered 8 species of tetrastigma in all the areas where we went to. I have seen all of the species. I think I have seen 10 species 11 out of 13. So I haven't seen the 2 species, but I've seen most of them. And in all these areas that we went to, we encountered 8 species of tetrastigma, of which 6 of them have been infected by Rafflesia. And this is a diagram of the host and parasite relationship. So these are the hosts, the Rafflesia species, and these are the host species. So this diagram shows you the S are those tetrastigma species that were not infected. So we also collected them in all these different areas. And what does it show us? It shows us that most tetrastigma host species are host to more than one species of Rafflesia. And also that one species of Rafflesia can infect more than one host species. And for example, the most common widespread species, Lagaske here, has at least 3 species of host of tetrastigma. So a lot of things are going on in this. So now we know that they're not, although the genus itself is very exclusive parasites of the tetrastigma host, you know, the species, they can host more than one Rafflesia. And that Rafflesia species can infect more than one host. And there's no evidence of host race formation in Rafflesia speciosa. For example, this is the species that is found in 2 islands. This is the only species that is found in 2 islands in the Philippines. And these are the 2 host species, Tetrastigma hammondii and Tetrastigma cf. Magnum. Also, the microsatellite data showed us that there are more than one individual plants of Rafflesia in one host. So you can even assume that one host has a population of Rafflesia just because, you know, and when you go into permitting, then this can be really difficult to explain because you want to have as many samples as you can. Yes. So that, and Rafflesia demonstrate that it may have multiple hosts. And this was the predictive power of host specificity was seen when we went to Mount Banahaw. Before we saw these senescent, you know, flowers, Mount Banahaw was only known to have one species of Rafflesia. But Blanco in 1880s already named 2 species. So there were 2 names. But then Merrill came in and it's like, oh, those are just synonyms of another species that was actually found in another island. I don't think Merrill saw, you know, all these Rafflesia. And so we resurrected 2 names of Blanco. And then we figured, you know, maybe we come back because we don't know what it is. Because what interested us is that this is the host of this common species in that mountain, Philippensis. This is a Blanco name, and Tetrastigma magnum. Whereas these senescent flowers are parasitizing this lowerite. So we figured this might be a different species. You know, we don't believe it's the same species. And then when we went back there, this is Lagaske. So Blanco gave 2 names within 4 Rafflesia in that mountain, Rafflesia Lagaske and Rafflesia philippensis. So those are the 2 names that we resurrected. And reproductive biology. We looked at all the species of Rafflesia and found out that Lagaske, Manilana, and Speciosa are bisexual. What does this mean? We also determined the sex of the flowers, whether it's female or male. And in these 3 species, some male and female flowers are actually genetically identical. So they came from the same plant. So therefore, they're bisexual, although some species are also unisexual. So if you look at this, this is the ovary, this is a female flower bud, and this is the male, the longitudinal section of the male bud, and these are the anther, the stamens. And there are 2 species of Rafflesia that I have discovered that have both the male and the female, but then we're not sure if both are functional or not, but at least there are 3 species. And finding Rafflesia fruits in the Philippines is very, very difficult. I think it took so many years before I found my first fruit. And they look like this. The fruit when it's young are very, very hard. You cannot, it's very hard. But then when it started to ripen, it's very soft, and they even try this. It's quite tasty. And there were so many hypotheses what could be the dispersers of Rafflesia, from ants to elephants to mammals to squirrels. And of course, elephants cannot be the dispersers in the field. We don't have elephants, although I hope we do. But in 2010, I think, we saw this. We saw this very decayed fruit. And then we also saw a lot of ants carrying seeds. We were lucky because we have a really good camera that would capture these very small ants subjects in the field. So this is an ant carrying a seed. And if you look at the seed, so this is a cross-section of the fruit, right? And these are the seeds. They're very, very minute. And this looks like a seed of a common ant dispersed, taxa with elaiosome. So it looks like a peanut, and we thought that one half of it has an embryo, the other is just for food. And conservation genetics. I'm going to go through this very carefully, very fast, because I'm running out of time. Microsatellite data. We did microsatellite analysis of 2 species, Lagaske and Speciosa. Lagaske is found in Luzon. This is the most widespread species from northern Luzon up to the Bicol Peninsula. We sampled the 14 known populations. These are disjunct populations. There are also populations here that we knew from photographs that we haven't sampled. And we also included one Manilana here because Merrill, this is the oldest name, Manilana from Samar. And then Merrill put all the small-sized Rafflesia that were discovered at that time as synonym of Manilana, so Lagaske and Philippensis. So we included that because we figure, it's morphologically similar. And what we found out is that there's no evidence of low genetic diversity, and also there's no evidence of inbreeding. So even though Rafflesia are quite rare, but they're still not in a very threatened state. And we found out that smaller populations have lower genetic diversity, except for one population. This is Lagaske from Mount Labo. Although we have a bigger number of population sampled, it has lower genetic diversity. And so this is the structure analysis, and we have 3 different clusters. From here, including this population, form one cluster. The Bicol Mount Labo population form another, and Manilana here is a different cluster. So it also shows the same in the principal coordinate analysis, 3 different clusters. And what are the implications for this in taxonomy? So there were 3 ways to resolve this. One, and we call this all the Rafflesia Manilana complex. You can perhaps recognize 3 species corresponding to those 3 clusters, or 2 species, Manilana and Lagaske, knowing that there's one cryptic species, or just one species altogether. We tend to go with this. Another species, again, this is the species that I said is found in 2 different islands, Panay, and 1 population in Negros. And unlike Lagasque Manila speciosa, there's recent gene flow among all the populations in Panay. They're very close proximity to each other. But then there's also a cluster here in Negros showing that there's not a lot of gene flow. And I think that's about it. So I thank everybody. I mean, in our paper, we have like almost a page of acknowledgement, but this is a result of almost 2 decades of research on Filipino Rafflesia. And thanks to the Marsden Fund, the National Geographic, and all the people and institutions who supported this research. Thank you.
Coffee break starts now, but we have time. So we have time for
Oh
questions.
no.
I'm gonna do it at the end. Actually, I think that's the right way. To
Okay, sure.
I have a question.
Oh no.
So do these, so the host trees that you find are affected by the Rafflesia, are they less fit than the species, members of the same species that don't?
I don't think so. The problem is, Nai said that I think lawyers kill their what do you call that? The customers more than the Rafflesia kill the host plant. No, we did not. Although it's very difficult to find flowering host plants when they are infected by Rafflesia, and also so I don't know if it's really true or not. But the hosts are very tall anyway, so it's really difficult.
Julie, any chromosome counting for this?
I think so. No, not for the Philippine species. Somebody would do that. Yeah.
Quick clarification. So are they monoecious? Dioecious?
They're monoecious, but imperfect flowers. How's that? Yeah.
Where are the tetrastigma species? Are any of those of like concern for conservation?
No, they're actually very, very common. So it's not. There's another group, one Filipino working as an assistant professor in the University of Long Island in New York, June Molina. They're now currently in the Philippines. They're trying to do ex situ. Good luck to that. But it's they're trying to collect fruits and some, because this tetrastigma can actually be grown just by cutting, so it's easy. But somebody is doing that.
The tetrastigma didn't seem to have the same geographic structure, given that it was matching up so many different species. So
yeah.
It doesn't have that same kind of limitation.
No. Yeah. What we noticed is that the most common tetrastigma in an area, for example, with Rafflesia, the most common tetrastigma species is the one that the Rafflesia prefers to infect. Although the less common can be infected in other areas where they're more common there.
Is there anything known about its ability to island hop?
The tetrastigma. I think so, but then the probability of the Rafflesia going with it, I don't know.
Does anyone study the ants?
No.
Because that seems to be like
no.
key to
I have a lot of friends who are ant experts. I said, please, you're teaching in this institution that is only 2 kilometers from so many Rafflesia, do something. But I think what he just did was the species of the ants that was based on our photos. He did not really do a lot of field work. So I hope they can do it. We can.
Is the host in Borneo the same genus?
Yes, the same genus of tetrastigma in Rafflesia. There's only one genus that's infected. Different species though.
Is it known how when the seeds are dispersed, do they land on the farm?
Nobody knows that. Nobody knows that. And if it is ants that are the main dispersers, then you would expect that, you know, that they have nests, and the nest should be adjacent or on the roots of these host plants.
All right, well, with that, let's take a break for the coffee break.
Bye.
We'll be back at 3:45. Yes, thank you. So hopefully you guys can hang out back there in Oxford. We've got a few talks to go. And for the Miami folks, please come back. We're going to do a photo at the end of the Hickeyites. And we got a little statement, things to read. So, and if you have to give a talk, still obviously come back. So. For those of you that are Hickey Lab alumni, associates in any way, stick around. Make sure you stick around all the way to the end because we'd like to get a big Hickey Lab family photo of all of those of us that have been here today. Otherwise, I'd like to get started with our afternoon session of talks. We have 4 talks for the afternoon, and then we'll have a closing statement here at the end. And starting us off is Laura Klein, who's going to tell us about 2 stories from lessons from. No, it's at the wrong one. Wow, it clicked on the wrong one. Let's see there.
We'll let her do that.
I saw learning floras and I went to it. There we go. All right, evolutionary insights from morphology, insights from 2 plant lineages. And here we go with the. Is Jim still there?
He's not.
All right. Oh, sounds like we've got the National Weather Service alerts going on. Flash flood warning. So.
Oh, is it?
Well, it rained as the local representative. It rained in the mountains. It's raining all over. That water runs down into the washes. So at the very least, it's flooding somewhere. With that, I'll let Laura take over.
Thank you. Thanks everybody for being here. My name is Laura Klein. I did my master's at Miami with Jim, and I'm going to try and talk a little bit about my master's and my dissertation work in a 15-minute talk. But the real reason I want to blend these 2 stories together is because it's all about morphology and it's all about hybridization. And I wouldn't be doing that without the man that we're here to honor today. So just to sort of orient us, plant hybridization can often be characterized by intermediate morphology and indications of disrupted meiosis. So of course they have to have an obligatory fern picture. And then indications of disrupted meiosis might be non-fertilized ovules in the top of the corn cob there, or these kind of crinkly, weird-looking little corn kernels. And of course, in spores, a good-looking spore should be round, full of oil, versus something that's kind of crumpled and sad-looking. So, what are the possible consequences of plant hybridization? So, this is just a figure I really like about how gene flow generates variation on which selection can act. So there are multiple different outcomes that can happen from gene flow. So this is from the mindset that you have 2 species coming together after being apart in secondary contact, whether that's geographic barrier or not. So you might have fusion of those genotypes. You could have an opposite situation where the 2 parental species come together and it in fact drives divergence further apart. Or you might have more of a blending in this hybrid zone contact or speciation of the hybrid individuals. And these differences are identified by careful observation of morphological traits. So introgressive hybridization specifically is the gradual infiltration of the germplasm of one species into another as a result of hybridization and repeated backcrossing, which of course was described by Anderson in 1936 in the classic iris story. And one of the best ways to assess whether or not you have introgressive hybridization is through a hybrid index. So parental species A would have all of their traits scored as a 0. parental species B would have all their traits scored as a 1, and then you add up those traits for all the individuals that you're assessing, and theoretically, your hybrid individuals will have an intermediate score. So what are the consequences of this introgression? That's what I explore in my 2 graduate studies. So I'll first tell you about what I did with Jim in North American Diphasiastrum species. So Diphasiastrum is this really cute lycophyte. There's approximately 20 species worldwide. It's a monophyletic genus. The ploidy level is 23. And it has these bilateral symmetrical branching patterns, these flat branches, and really cute little microphyls. Kind of looks like something that should be found under the sea, but it actually lives in more northern temperate zones. And it has these underground gametophytes. So the 3 species that I focused on for my thesis work was Diphasiastrum compinatum, which has that more circumboreal distribution, Diphasiastrum digitata, which is primarily all in the northeast of North America, and then Diphasiastrum tristichium. All of them are very similar in their morphology, but we can tell them apart with morphological characters. And there have been described hybrids between all these different parental species. So Barricundum is a hybrid of digitatum and compinatum. Xylori is a hybrid of compinatum and tristichium. And then Habertii is a hybrid of digitatum and tristichium. So what are the possible consequences for these intermediate forms? The goal of my thesis work was to determine if clear boundaries exist around 3 species of Diphasiastrum and investigate the role of hybridization, with the hypothesis that these 3 species represent variant forms of a single morphologically continuous species. So we would then predict that if it were one species, a continuous frequent morphological variation would be observed. Intermediates would be as or more abundant in extreme forms, and they would be fully fertile. However, if these are 3 species undergoing hybridization, then we would predict that intermediates are less abundant. Intermediates would be in areas of sympatry, and there would be reduced fertility. So I started with 1,423 herbarium specimens. I selected from those roughly 25 accessions from each parental taxa that represented just good morphological characters and were representative of their distributions out in allopatry. And then I selected putative hybrid taxa that were found in sympatry and had been described as hybrids previously. So I started with a data set of 34 initial characters and used 15 for my analysis, of which 4 clearly distinguished all 3 parental taxa from one another, and 11 were significantly different from 1 or 2 of the hybrid taxa. So from this, so I had this beautiful grand plan that I was going to show up here and be like, you know what, Jim? I finally published my thesis. Here's my copy. That didn't happen, but I did make new PCA plots. It's pretty embarrassing what I did for my thesis, but these are really lovely summaries of the variation described by those 15 characters. So in the blue, we have Diphasiastrum complinatum, and the yellow here is tristichium, and then in the red is digitatum, and there are 95% confidence ellipses around those points. And when we start to then layer in the different putative hybrid taxa, we find that the putative hybrids fall between their parental taxa. So in total, that's kind of a big jumble. I just kind of like to show it all in one. And we also did a hybrid index, and so I won't go through all of it, but just to say that on the left-hand side of the figure, the 0 is one parental species, and then the higher number is the other parental species. And you're seeing that black bimodal distribution for those parental taxa measured, and all the hybrid taxa fall in the middle. So they're all intermediate, which is not what we would expect if this was one continuous species. We would rather see more of a homogeneous distribution of these traits. So I didn't have time to tell you about that we also did some abundance estimations, as well as some spore fertility assessments. But what we find is that, in fact, interaggressive hybridization is occurring among Diphasiastrum componatum, digitatum, and tristachium. And the intermediates were less abundant. Intermediates were in areas of sympatry, and there was reduced fertility. So for then my dissertation work, I really wanted to stay working in morphology. I really wanted to continue working in hybridization. But I moved to angiosperms. And I thought maybe I would get some booze at this point, but I just wanted to say that even Jim likes angiosperms. So why grapes? Not only did I learn from Hardy that I really enjoy ethnobotany and economic botany, but grapes are just an excellent system for studying hybrids because hybridization is common both in nature and in cultivation. So Vitis vinifera is the wine and table grape. It's a single species from which there are over 1,000 different cultivars. And then Vitis estivalis is a native North American grapevine. And if you hybridize these together, you get the Norton grape. It is a wine. I'm sure none of you have heard of it because it's not that good. But it is the state grape of Missouri, so I would be remiss not to mention that. And that's where I am now. So hybridization is really common in cultivation, but it also is extremely frequent in nature. So as far as we know, grapes are completely interfertile, which is a very interesting evolutionary question. How can that be? And one of my favorite viticulturalist botanists from the turn of the century, T.B. Munson, said, "It was at once apparent to me that a botanical investigation of all species of our wild grapes must be made before much valuable work in this field could be done." So how does naturally occurring hybridization affect Vitis evolutionary relationships? Vitis is a great system to look into this because there is so much variation in Vitis already that has been characterized. So in cultivated grapevines, the discipline of amphilography was created to describe variation in grapevines because grapevines have a homologous leaf shape. Generally, they're always cordate. They have this palmate venation. There is some degree of lobes and sinuses, both at the proximal and distal ends of the leaf. So it's just a really great system to look at leaf shape in general. And even better, where I live in Missouri is in the hotspot of Eastern North American Vitis diversity. Vitis has 60 species globally, of which 13 approximately occur in the Midwest. For my focus, I picked two closely related species. This is a phylogeny. I did not put the citation up, but this is from 2008 in the American Journal of Botany, or 2018. And I focused on close relatives Riparia and Rupestris. So one is the riverbank grape, Riparia. It's your traditional grape. It climbs up trees. It's a liana. It has these nice cordate leaves, and it occurs all across the northeastern United States and North America. And I also focused on the rock grape, Vitis rupestris, which has a little bit of a zone of contact down here in Missouri and also along the Ohio River. It occurs in these rocky creek beds and has this more reniform leaf shape. So my questions were, what is the range of leaf morphological variation in natural populations of Vitis? And does leaf morphological variation correspond to known species identifications, to geographic origins, or both? Oh, I should have fixed that. So we looked at 14 sites throughout the Midwest to represent a latitudinal transect of the Vitis riparia and Vitis rupestris distributions. And again, it would be great if I could just collect Riparia and Rupestris and Allopatry and in Sympatry, but there's 13 other Vitis species that could potentially occur there. So at every site, I collected at least 20 accessions of my target species and then anything else I could find, with a particular eye for any hybrids, of course. And to analyze leaf shape, I use digital morphometrics, and so I'm going to go through it very, very briefly. I would collect all my accessions from the wild, I would image these, and then use landmark analysis to characterize leaf shape. So I used 17 landmark points to highlight aspects of the leaf shape like sinuses, lobes, length, width, and then also venation patterns. And when you landmark, you're essentially creating an X and Y coordinate. And that X and Y coordinate can then be mapped. And you can do what's called generalized Procrustes analysis on it. Generalized Procrustes analysis is just a sophisticated math algorithm that allows you to compare shapes without worrying about size. It scales the size so that you're just comparing your shape. And then you can summarize that variation using principal components analysis. And so principal component analysis, or principal component 1 summarized 44% of the variation in our data set of 414 individuals and over 12,000 or 1,200 leaves. And so what we see for the mean eigenleaf, it's sort of that traditional coordinate leaf shape, but then on the plus minus standard deviation, we have leaves that are wider than long versus longer than wide. And we do see that there's kind of a standardization. It's very close to the mean leaf shape for a lot of Vitis species, but for things like Vitis rupestris or Vitis vulpina, it's a little bit more extreme. And to look at this another way, all of these points, the colors represent species, the points represent leaves. On the x-axis, we have populations. On the y-axis, we have the principal component 1 score. And interestingly, we see these shifts. So in populations where there are a heavier community of Vitis that exists, Vitis rupestris and leaf shapes starts to kind of go lower into principal component space compared to where it is in other distributions. That's very interesting. And this makes me think that there could be some evidence for a selection against hybrids. And I don't have time to talk about the genetic data that I did for this, but out of 414 individuals, I only found 3 hybrids, and I was looking for them. So, and this is a species that, or a genus that's supposed to be completely interfertile. So I am excited to hopefully investigate that a little bit further. So from this, we find that leaf shape primarily corresponds to species identity and somewhat with geographic location, and that the range of leaf morphological variation in nature in natural populations of Vitis may be affected by selection. With that, I have to thank the Hickey lab. I was there from 2010 until 2012. And then I moved to another Miami alumna's lab, Allison Miller. But I just, again, I'm forever indebted to Jim. He was the person who challenged me to think about things and ask good questions and inspired me to continue to do the things that I do. So thank you, Jim. You're the best. And thank you all for your attention.
While we make the transition.
Yes.
Is that close to Potterai in the first picture?
I'm sorry?
Is that close to Potterai?
All right. So Melanie, we can see you now. You're on the big screen, by the way, because all of us are doing PDFs. Well, I think those of us with PDF, she's going to be there. So I emailed her to turn off her. Yeah, you're a PowerPoint, so you're okay. All right. Well, I'd like to introduce our next speaker here in the Hickey Fest. Today is Jessica Budke from the University of Tennessee. She'll tell us about exploring relationships with moss genus dissidents using molecular data generated from herbarium specimens.
Thanks, Mike. Thank you all for being here this afternoon. I was an undergraduate student at Miami University in Jim's lab, and as many of you know, I took introductory biology from him. As many of you know, he's a very enthusiastic instructor, so I went to his lab excited about doing undergraduate research. And Jim, of course, sent me into the field. So he sent me to Tennessee to hunt for a new species of isoetes that they had sort of seen down there, and I got a lot of really great experience working in his lab, learning how to pickle isoetes, how to make herbarium specimens, how to section and squash chromosomes. And ultimately, this resulted in my first 2 scientific publications describing a new species of Isoetes tennesseensis. I fell in love with Tennessee out in the field, and as Mike mentioned, I'm now at the University of Tennessee. So I'm the herbarium director there. I also teach field botany, and I was inspired by Jim, and I try my best to share my excitement for plants with students and teach them about the importance of learning how to identify species so that you can find those new species when they're out there on the landscape. Fortunately, though, I did not move into flowering plants, right? So I don't study things with flowers, but instead I study bryophytes. So my research is very similar to things that are continually happening in the Hickey lab. I study structure, function, relationships, morphological evolution, and it really centers on using biodiversity collections. Some of the main features to tell apart mosses are often based on a combination of sporophyte and gametophyte features. Mosses are often thought to be morphologically austere taxa. They're very small, they don't have a lot of features, but there's actually more to be found than you might imagine. So you just have to look a little closer and bust out your hand limbs. The genus that I'm currently working on is the moss genus Physidens. There's a single genus within the family, Physidendaceae. These are very distinctive mosses. You too can identify them in the field. Most mosses have spirally arranged leaves that come out of the stem. Physidens are disticus, so they have leaves that stick on the left and the right side. They're pocketed together, so the common names for these mosses are the pocket mosses. So essentially, if you put them sideways, they'll look nice and flat. But at this level, you can identify them to genus. Getting them identified to species can be much more challenging. So in this group, there are really robust morphological hypotheses that are defining a number of subgenera and sections within the genus. There's two main hypotheses that were proposed in the mid to early 2000s. One by Purcell and Brugmanega, based on mostly European specimens and North American specimens, with a perspective there. And then Suzuki and Iwatsuki sort of came up with another classification system based on primarily Asian specimens. So it sort of sets up really interesting sort of sectional hypotheses that we can then test with additional data. And as you see, some of the sections are sort of equally defined in these two systems. Some of them, there's a little debate about which level this sort of group should be at. And then we have our classic splitters versus lumpers in terms of how you think about the relationships between the species. So we set out to really sort of examine the classification system of Purcell and Brugmanega, and all of these classification systems are really based on anatomy. So this is a transverse section through the midrib of the moss, so through the sort of costa anatomy. And there's sort of three main groups of cells that you can see here. There's two steroid clumps, there's a central cell, and there's a couple of guide cells to sort of define this bryoides type that is representative for the three bottom subgenera of Physidens. The taxifolius type has sort of a range of guard cells around the costa, two clusters of those steroids, and multiple central cells, unlike the bryoides type that only has one. And this defines both section Pachyphysidus and section Crispidium. And then finally, there's a third type, the oblongifolius, which has three clusters of these steroid cells, one group of guide cells, and then also a number of these central cells. And so this sort of anatomy, which mosses do have, is used to define the sections in addition to a number of sporophyte morphological features. So sort of it's this broad combination of sporophyte and gametophyte morphology that have been used to define both of these two systems for classifying the genus Physidens. So our initial goal was to basically test these morphological based subgenera and sections to see if we could recover them using molecular data. Some of the other hypotheses that are going on within this group that have been defining these sections are the difference between Pachyphysidens and Amblyophalia. One is defined by having large gametophytes and monoecious gametophytes. Others are defined as small in Amblyophalia, and they're both monoecious and dioecious, so we have these differences in sexual systems. And so one of our big questions is, where is this size cutoff? Like, what defines a big moss versus a small moss? And are all these species that have similar sexual systems each other's closest relatives? Or do we have these multiple transitions in sexual system across the phylogeny? Further down here, examining the section Physidens and Sarawaki and Octodoceras. Some of the sections are completely terrestrial, while others are aquatic. And one of the questions we have here is asking, are these aquatic species actually each other's closest relatives, or do we have multiple transitions from these terrestrial systems into aquatic habitats? And then finally, the subgenus Aloma is a really widespread tropical subgenus that are highly morphologically diverse. And so our questions focusing here are whether these tropical species are actually closely related to each other, or are they just morphologically similar, or are they morphologically similar to temperate species? And so do we actually have a tropical clade that's diversified in place, or are there different evolutionary scenarios happening? So our initial sort of goal was testing these morphology-based subgenera and sections. We're using DNA sequence data. This is sort of an ongoing research project in my lab, so I'm going to be showing you sort of some of our preliminary data. All of this is based on using herbarium specimens. So we're extracting all of our DNA from herbarium specimens, and one of the things I like to sort of talk about and pitch to the students is that all of our herbarium specimens were identified by all of the experts that did these sort of subsection and subgenera classifications. And so we're really collaborating with researchers of the past to sort of come back and test their hypotheses using their own collections, which I find to be really fun. And it gets the students pretty excited to think that they're sort of part of this process. So we targeted 3 loci because this was basically an undergraduate sort of focused project where my students were generating data using Sanger sequencing. And then we built a molecular phylogeny. So this is sort of the broad scale phylogeny. The family Physidens, or the genus Physidens, and the entire family always comes out as monophyletic and very strongly supported. And we have reasonably strong backbone supports for some of the major clades that we're seeing here. So we have both maximal and strong support with the darkened branches and the dots. If we zoom in and take a look at this crown group in the phylogeny, we can sort of start to address and examine some of those questions that I posed at the beginning. So one of the ideas was looking at these different sized gametophytes and these different sexual systems between section Pachyphysidens and section Amblyothalia. And what we find is that all of the section Pachyphysidens individuals are coming out in this crown group and are reasonably closely related. And then we have the smaller gametophytes with this mixed sexual system that are coming out in 2 clades that are not very strongly supported, mixed in with some other species that are coming from a completely different section. So, I mean, overall, it does look like we're getting good splitting between these 2 sexual systems, with maybe some species that are a little more variable in between. When we look at the question of terrestrial versus aquatic species, we were sort of asking whether they're transitioning multiple or single times. And we find this large clade of terrestrial Physidens and sort of a single transition into the aquatic habitat. However, the aquatic species are much rarer, they're much smaller, they're sort of harder to get a hold of herbarium specimens. And so we really need to sort of increase our sampling in these aquatic taxa to get a sense of whether or not they're coming out in multiple locations. But at least here, 2 of the species are coming out as a monophyletic group. Another of our questions was looking at these tropical species to see whether they were closely related in subgenus Aloma. And we have this large clade of members that are in subgenus Aloma, but then we have some individuals that are sort of popping out in these other places in the phylogeny. And if we think about where some of them are from, so Physidens paperculus, this is a specimen from the United States, from California, and it's sort of nested within this larger clade of species that are all California endemics. So we have some interesting biogeographic patterns where these tropical species are actually more closely, or these species that have been grouped with these tropical species are more closely related to temperate taxa. And if we look over here, we have several species that are from Japan and French Polynesia that are sort of nested within this larger clade of species from Australia, Asia, and the Pacific Islands. So we may have some interesting biogeographic patterns to sort of tease out that may be important to expand our sampling in some of these species in order to look at the relationships further. And so this is sort of the broad picture of how those different subgenera and some of the questions sort of fall out in terms of the morphological evolution. And so where we're headed next with this project is that we're improving our backbone resolution. So we have a number of specimens and data that's come back from next generation sequencing. So you might have seen Matt Johnson's presentation looking at, they're using a bait set for 800 bryophyte genes and also assembling the chloroplast and mitochondrial. And we've been working with their targeted bait sets to see if they'll work on Physidens. We also have some of the great GoFlag data that Emily and Stuart's group have been generating. And so we're going to start to analyze that data to really get a sense of how much additional support we could lend to this backbone with some new sequences. And we're also expanding our sampling. We have some subgenera and sections that are unsampled, and so we'll be broadening our geographic coverage to sort of increase, hopefully, the resolution that we see in the phylogeny. Because this family has about 450 species, and so we have about 50 taxa in this tree. And so we really need to sort of increase our sampling to see what's going on. And there's also other folks around the world who are interested in fissidens. And so this is the molecular phylogeny that came out from the group in Japan. So they were sort of out to test that other hypothesis looking at the relationships among the Asian taxa. And so we have a nice sort of comparative system to look at their hypotheses focusing on Asian taxa, our sort of more broader North American, Europe, worldwide sampling, and see how those two line up in comparison. And with that, I would especially like to thank the herbaria that let us destructively sample some of their specimens. I had a number of student collaborators who worked on this project, and I don't think I would have been able to do and sort of be where I am as a scientist without Jim and other mentoring support, and then my funding. And then I'll take any questions.
Here's time for a question or two while we transition.
Your Fissidens ventricosus, we call that in the best, very distinctive aquatic complex.
Oh.
So check that.
Yeah, so going back to and I think we need to be sure we get a firm classification, because I think one of the other things is getting down to actually the specimens themselves and seeing for individual specimens, because some of the fissidens can be more facultatively rather than obligately aquatic, and so I think we need to know exactly where each specimens come from to categorize it as aquatic, terrestrial, or facultative. That's a good one.
We know it's just absolutely rocks on the street.
Okay, yeah, we'll be sure we.
If they're not on rocks, you don't know.
Thank you.
All right. So I get to moderate myself, I guess. No, it's not entirely. So for the next 3 hours now. Well, I'm happy to, as the local host of the meeting, also happy to welcome everyone here to Tucson. It looks like we got a little bit of sort of monsoon rain today. But I'm going to talk about a case of potential homoplate and allopolyplate hybrid speciation here in Selaginella, the Sonoran Desert. But first, I want to just mention sometimes with Jim Hickey, who I did my master's with back in 2001 to 2003. This is from the Bahamas trip. Jim wanted to be taller than me, and so forced me to crouch down. Yeah, anyway, and here's another one of these famous Jim with the snake pictures that Mike Vincent was showing earlier. I think this is a brown racer out there on the western edge of Andros Island, you know, where it kind of dwindles into the Sargasso Sea out there. And here we are with Jim, time we forced, our lab forced him to go to Botany 2003 in Mobile. And we took the fern field trip picture that time, and he had to be right in front. It was pretty funny. So, and one of the great things I enjoyed during my time working on revisionary work, revisionary taxonomy in Jim's lab, was diving into these species complexes, right? I think a lot of us that get into studying ferns and lycophytes in particular really dig all the hybridization and polyploidy that happens in these groups. And this is the North American Dryopterus hybrid complex. And one of the things you notice about these reticulograms when you look at them, there are a lot of diploid species here that produce fertile polyploids of some sort. There's a lot of sterile diploid hybrids, or you just don't really see the diploid hybrids anywhere. And this is a pretty common thing out there in nature that we have. Maybe estimate about 15 or 20% or so of extant species in plants are allopolyploids. And there are very few known diploid hybrids, despite that abundance of allopolyploids out there, the fertile diploid hybrid species. And so one of the things that there's a whole host of questions that we want to ask about the biology of polyploidy and hybridization. But one of the things that we often want to know is what are those relative contributions of polyploidy and hybridization to plant evolution? And one of the challenges in teasing apart those different forces is that many of our allopolyploids are actually hybrids also. So you get into these sort of what is responsible here? What is for some of these phenotypic changes or the ecological changes that we see? Is it hybridization, or is it polyploidy, what. And so one of the things that we'd really like to have, to help sort some of this out as plant evolutionary biologists are examples maybe of where we have both diploid hybrid speciation and allopolyploid speciation from that same parental cross, where we get the same combinations to try to see how that evolutionary experiment's played out in real time and in nature. And these are kind of few and far between, actually pretty rare. There are a couple of potential examples in the literature. The most compelling is our most Gottlieb's work in Stephanomeria, the diploid hybrid Diogenesis and its tetraploid Elata, which appear to have perhaps the same patterns when you look at isozymes and ITS data. And another potential example in peony, it's a little bit less compelling of a case that those two are from the same parental cross. But they're out there nonetheless as potential examples. And what I want to talk about today is a potential example here in the desert, what I call these unicorns of plant evolution, where we might have a double-ploid hybrid and the allopolyploid from that same cross. And this is work that's been done by one of my PhD students who's now recently graduated, is at Yale as a Donnelly postdoctoral fellow working. He's currently collecting viburnum, I think, for Michael Donahue and Erica Edwards. He totally does not approve of me putting his picture with these unicorns. And I totally tweeted that out to a few thousand people. So Selaginella are heterosporous lycophytes, about 800 species, and they're renowned for many of the species are resurrection species, including these. They have really small nuclear genomes, and that allows us to do some interesting work with it. And the two species in question that we've been studying out here in the desert are Selaginella arizonica, and it forms these big mats across the desert. It's out here all over the slopes. And actually, in ecological surveys of the Sonoran Desert, anywhere from 30 to 40% of most of these surveyed sites are actually, the ground cover is actually Selaginella of some sort. Like for example, at Tumamoc Hill, 40% of the ground cover there is actually Selaginella arizonica. So it's not a rare plant out here. You can almost do transect for these species in particular, get out of the car, go walk over and grab some, and not do anything because they resurrect. So for doing genomics, it's really quite nice. This is another diploid species, Selaginella aeromophila. This occurs in Southern California and in the Baja. This is from Anzo Borrego, a wonderful site called Hellhole Canyon. It's not that bad, but anyway, these are these two species, and I don't think it's going to play our little resurrection. Well, you can see it on my lab's website, a video of these resurrecting in about 3 or 4 hours when Hurricane Newton came through back in September 2016. As I said, these two diploid species of Selaginella occur in pretty distinct habitats. Selaginella arizonica over here in the Sonoran Desert and occurs throughout Arizona. Aeromophila is found in much lower elevation sites over in California and in Mojave. And they have these hybrid zones here. They have some contact zones, I should say, in Oregon Pipe National Monument and outside Phoenix and places in rural Maricopa County. There are strikingly different looking for Selaginella, and I know that the angiosperm folks in the room are going to roll their eyes. But they're actually pretty easy to identify. To tell these two species apart, at least. Aeromophila is much smaller, more compact, as you'll see. And they have these contorted pinnae on the top of the tips of the microphyls, whereas Selaginella arizonica, and these are very deciduous. Whereas arizonica has these really solid, strong, like they'll poke you, indurate tips on those microphyls. And as I said before, these two species have a sort of a hybrid zone that we've been studying out here at Oregon Pipe National Monument and in Phoenix, and it does continue in the proximal areas of Mexico. We don't have a lot of collections from those sites, or in our collections at least. And when you start looking at herbarium specimens of these plants, when you go out to plants that have been collected in places like Oregon Pipe, like this specimen here, you'll see that it was first identified as Selaginella arizonica, right? And then here it's been relabeled as Aeromophila, and then you can't see it because it's covering it up. And it says, wait, this is mixed. And then there's another thing that says, wait, these are hybrids. So there are whole pools of these, for those of you that spent your time in a herbarium, where there's just piles of these intermediate specimens, where Mike Windham and George Yatskovich have gone back and forth, I think, on labeling some of these. So when you look at the morphology, you can see here that Aeromophila is a much more compact species than its diploid relative arizonica. And Anthony went through and collected a number of traits, about 18 traits from about 100 sheets. from both Aeromophila and these two species that we believe are hybrids growing here in the hybrid zones from Phoenix and Organ pipe, which are distinct from each other as well. When you analyze the PCA of the traits, Anthony finds that these are intermediate between the two parents, as you might expect, for hybrids taxa. And when you do the chromosome counts, you see that it looks like the plants around Phoenix are always about double the two parents. So the two diploids that we have in this case are 2n equals 20, and Aeromophila is 2n equals 18. And 2n equals maybe 38 and 18 in the case of the organ pipe plants. This is a range, not because we think the actual number ranges. These nuclear genomes are about 60 to 80 megabases. The chromosomes are about 3 megabases long. They are the hardest things in the world to count. So the length of a Selaginella chromosome is about the same length as a human haplotype block, right? Tiny little things. So we know that they're morphologically intermediate. We know they're out where they are in the landscape. And we know that there are a couple ploidal levels at play here. What we really want to know is, are they actually hybrids of these species? And if so, is there evidence of hybrid speciation happening in this case? And to explore that, we've sequenced and analyzed annotated genome and transcriptome sequences. And we've looked at testing their hybridization status and the relationships using model testing in the phylonet framework, as well as using the D statistic, in particular the D1 statistic for Pibbins and Hahn, to test for evidence of hybrid speciation. I'll show you some results from that work here now. So just think about this in a very general framework. Of a hybrid is descended from two sampled parents, and you sample a whole bunch of gene trees. You'll get some gene trees that follow a pattern like this, where you have one parent Arizonica that is sister with a hybrids taxa, whatever it might be, sister to it, maybe 40% of the time, 40% of the gene trees. And here, Selaginella wallisii is an outgroup that we use in this case. And maybe some other fraction of the time, you'll see that the hybrid taxa goes with the other parent. And you always end up with some fraction where wallisii is in there, and you get some different set of trees. And with phylonet, we can also not only test those kinds of models, but test the support for phylogenetic networks. And what you end up producing are figures that look like this, where we ask, are these actually the parents too, when you combine all those information? And you get different kinds of inheritance probabilities because of all sorts of integration and backcrossing, as well as the fractionation and diploidization that might be happening depending on the ploidal level of these hybrids. So what do we see when we look at this in Selaginella, these Selaginella hybrids, or putative hybrids? So this putative homolipid hybrid organ pipe and this allochiploid phoenix. So looking at organ pipe, analyzing about 300 single copy gene trees pulled out of the annotated genomes and the transcriptomes from these populations. We find that about 48% and 52% of the genome comes from Arizonica versus Aeromophila, and that we do find the best supported model is this phylogenetic network with the highest log likelihood score. For Phoenix, we have a similar number of gene trees. We find a similar ratio, about 40% of the gene tree of the genomes coming from Arizonica, 57% from Aeromophila. And again, both of these are we've tested over 14 different combinations of phylogenetic tree models. And these are the best supported models in the situation where these are hybrid products of Arizonica and Aeromophila with the highest log likelihood scores and really high delta AICs for adding other. So once we add any particulate edges, we get much better model support, for example, for these particular sets of relationships. So that's pretty good evidence that these are in fact hybrid products of those two parent parental species, or at least we're really close relatives of those parents in our sample data set. How's that? But we believe these are probably the parents based on some other data too that we don't have time to talk about. But so we do see that there's pretty good support for this, but that doesn't tell us is it hybrid speciation, right? That's a very different thing. And so with this sort of data, with phylogenomic sorts of data sets like this, there are some new ways of testing for support, at least, or if the pattern that we see in the data are consistent with hybrid speciation. And one of those is this new D1 test statistic in D2. We're just going to talk about D1 from Mark Hibbins and Matt Hahn. This was published in Genetics, I believe, back in May. It's been on bioRxiv for a little while. And the way this works is that you can imagine that instead of having hybrid speciation, we could have hybridization and speciation don't happen at the same time, right? So it could be that in this case, we have here, for example, Arizonica, and maybe this organ pipe plant diverged more recently than Aeromophila. So our coalescence time doesn't go back as far. And then subsequently, this taxa hybridized with Ermophila, giving you a signal of hybridization in the data. Alternatively, you could have hybridization and speciation happening at the same time, right? In a situation that we would expect with hybrid speciation. And the D1 statistic allows us to test for this by looking at the difference in divergence and the coalescent times from Arizonica minus Ermophila, in this case, subtracted from Ermophila minus organ pipe. And if you have that D1 doesn't equal 0, then it tells you if it's significantly different from 0, that hybridization and speciation are not, were not coincident. How's that? Or not, we're far enough apart in time you can tell them apart. And if you have hybridization and speciation, D1 is indistinguishable. It's approximately 0. Those two processes are indistinguishable, and it's consistent with a hybrid speciation event. And that's what we saw here. So we saw that D1 was 0.0295. That's a non-significant p-value, and that D1 is indistinguishable from 0. This is exactly what we'd expect with this statistic. This is a case of homoplate hybrid speciation. And so overall, across these data sets, the sort of phylogenetic analysis of hybrid speciation, we do see that these two taxa at organ pipe and Phoenix are likely the hybrid progeny of Arizonica, Selaginella Arizonica and Ermophila. And there are different ploidal levels, suggesting this might be a really nice worked out case, hopefully, of homoplate hybrid and now polyplate speciation from the same time. And in the case of organ pipe, where we can do this multi-species coalescent test, the D1 test, we see that hybridization and speciation occur at the same time or indistinguishable from those two, based on that statistic. And Anthony is doing tons of other data here, whole genome analyses and population genomics that they don't have time to talk about, ecological, ecophysiological analyses, and they all resurrect in different ways, and there's all sorts of fun stuff that he's got a stack of papers to write in between all his viburnum papers he's going to be writing, no doubt. And with that, I'd like to thank everyone for coming out today and take any questions.
I saw there's a 5% indifference in the genome composition between the two hybrids.
Yeah.
What happened?
I don't know off the top of my head. We haven't looked at that. It's from this subsample. It's you know, in these analyses, it's always slightly different. Like phylonet doesn't exactly tell you what those genes are. So but when you look at the two genomes, it's not biased in any overall kind of way if you think about genotologies or anything like this. And it looks like there is a lot more, clearly it's sort of biased towards Ermophila. In the population genomic data, it doesn't look like there's any more like gene flow that way or anything driving it. So yeah, I'm really excited to see that once we get those next set of analyses done. So, yeah, and to see that in the population data too is what's sort of moving and what isn't. So yeah. All right. Oh yes, by the way, real quick, and then I'll let Lee go. We have no more talks, so Lee's after this, so then he's got a lot of time, so we can have a minute. So
so hybrid should have two alleles, one from each parent, but it seems like you got either one.
I'm sorry, what?
The hybrid should have two.
Yes,
alleles, but
it does.
You got either one from either one parent.
Oh no, it has. So when we make those, like in the phylonet gene trees.
Yeah.
Oh yeah, so that's sort of showing that you'll get one set of gene trees that show this and one set of this, and sometimes that's allelic and sometimes that's not, right? Because some of the alleles get replaced or or removed. I don't know how many of the sites in the genome are still sort of or segregating alleles from both parents, but you do some fraction of those is that for sure. Yep. All right. I'm not going to take up any more time. I told Lee I would just keep talking over. That's the danger of having your student follow you. So Lee is a double dose of Hickey because Lee was a undergraduate at Miami, and now he's my student. So he gets Jim in two different ways. And Lee's going to talk about his recent work on why homosexual ferns have high numbers of chromosomes.
All right, thank you all for coming here. Such an honor to speak at this section. So I was an undergrad at Miami, and I worked in the Hickey lab, and I also work at the herbarium with both Jim and Mike. So it really is Jim and Mike that bring me into the world of plants. And as growing up as a botanist, I really appreciate the morphological diversity, all the species And then when I came here for graduate school, I started become more interested in this pattern, which I think is one of the coolest things that we can see. So chromosome number variations. So just to represent a few extreme cases in here, all the flying plants have n equals 2, including one of the haplopeppas, which our lab is about to sequence the whole genome. And then if you compare that, the Ophioglossom reticulatum, which have n equals more than 1,000. And if you don't know that plant, right, Ophioglossom reticulatum, which has the highest chromosome count among all the eukaryotes. So chromosome number variations. Here I want to just a few figures to show you the variations among 3 major lineages: fern, lycophyte, and eudicots. So 2 major take-home message from here. So on the x-axis, it's showing you the haploid chromosome numbers. So one thing really cool is if you look at in ferns and in lycophytes, the number goes way up, goes more than 100. If you look at eudicots, it's less than 100. Very few species go up more than 60, 70s. So that's one. And then if you look at the y-axis, it's showing you numbers of species. So how many species that has that haploid chromosome counts. So in flowering plants, when eudicots, here you see the most common number is 9, 11, and then you see lots of spikes suggest there's lots of aneuploidy, that means individual loss and gains of chromosomes. So you see lots of aneuploidy in flowering plants. But if you look at the patterns in fern and lycophyte, you don't see much of that aneuploidy. It's much more rare. You don't see that many spikes. And if you look at ferns closer, most common number is 41, and then 82 is also really common, 63, 36, 72. So that's really different compared to flying plants. So this the pattern has been sort of noticed for a long time. One of the classic work from Kalkowski and Baker, 1966, what they found is Homosperous fern has way higher chromosome number compared to both angiosperm and heterospore sperm. So for those of you who are not familiar with this term, homosperous just means they produce single type spores, become typical bisexual gametophyte, produce both egg and sperm, and heterospores, which means they produce mega and microspore, which turn into female and male gametophyte, which produce egg and sperm. So pattern has been found, but how do we explain that? So there are multiple explanations out there. First one proposed by Duncan and Smith in 1978. So it could be that fern just have really high ancestral chromosome number. Well, throughout our analysis, so we have a chromosome number analysis across the fern phylogeny suggests that's likely not the case. So on the axis here, we're showing you the distance from the root towards the tip of the phylogeny. So this is the root, this is going up to the tip. On the y-axis, we're showing you ancestral chromosome numbers. So you can actually see there's multiple increase, right, when you're going up to the tip. So if it's ancestral high chromosome number, you probably should see an opposite pattern. You should start from somewhere high. Well, there's another alternative hypothesis that you can see from Hoffler and Solis in 1986. It's the ascending aneuploidy. So in flying plant, we know that there's lots of aneuploidy out there. If there's lots of aneuploidy, this is what you expect to see. You see lots of spikes on this kind of figure. But in fern, we don't see that. Aneuploidy is much more rare. Most of the things have just this few numbers. We don't see that many aneuploidy out there in ferns. So this leads us to the leading hypothesis. It could be due to multiple rounds of ancient whole genome duplication or paleopolyploidy in fern that caused them to have that many chromosome numbers. So Hoffler and Solis 1986 and Hoffler 1987, they first proposed the idea that fern may have experienced multiple rounds of ancient genome duplications, followed by gene silencing, but not chromosome loss. And Barker and Wolf, 2010, further asked the question, so they compared that to angiosperm and asked the question, so why do homosperous fern have so many more chromosomes compared to flying plants? Especially we know that in flying plants, there's tons of, they have a really rich history of ancient polyploidy. So this is our current analysis, current result from the 1KP study. We found tons of ancient whole genome duplication across the phylogeny of angiosperms. And we estimate, well, more than 75% of the flying plant family has at least one ancient whole genome duplication. So going back to our question, so why do Homosperous fern have so many more chromosomes compared to flying plants? Well, we have two hypotheses. Hypothesis number one, well, it could be that Homosperous fern just have much more rounded ancient polyploidy so that they have more chromosome numbers. Or Homosperous fern, they may have similar or even fewer rounds of ancient polyploidy, but they just don't lose their chromosome, and that causes they have much more higher chromosome numbers. So to test those hypotheses, we need to further ask two more questions. So why, where in the fern phylogeny did this ancient genome duplication event occur? And then what are the frequency of ancient genome duplication in both fern and flowering plants? So to test that, we assemble a pretty big data set, including 157 fern and lycophyte transcriptomes. A lot of them coming from our 1KP project, and also the Shen et al. 2018 paper also have lots of fern transcriptomes. So we put those data together to do this test. So how do we infer them? So we have our pipeline called DoPipe, which is available in our evopipe.net. So for those of you that are not familiar with this analysis, it's also called KS-plot. So we take individual transcriptomes, we generate gene family phylogenies, we use PAML to calculate the node KS, and then we plot all the node KS into a histogram. And if a species have ancient whole genome duplication event, you would expect to observe this kind of peak in the histogram. And if a species don't have whole genome duplication event, for example, this is a histogram from Sonagulata, we know that they don't have ancient polyploidy. You will not expect to observe those peaks. So just give you a few empirical results. So we do observe evidence of ancient genome duplication event in Equisetum. We also see that in Magradia. Basically everywhere we see that in Xylodon, 2 rounds of genome duplication in tree ferns, and also in Pteridium. And if you look at a lot of them, right, sometimes you will encounter this kind of examples. So here, this is the species tree with 5 different taxa. A, B, and C are close related. You sometimes see there's peak at the similar regions. So is that due to multiple independent ones, or it's a one shear event, right? So we need our next analyses. So to sort of confirm that. So we use our the algorithm that we developed in our lab called multi-taxon paleopolyploidy search, or MAPS. So we take multiple transcriptomes, we do protein predictions, BLAST-P, and then we use AlphaFinder to cluster gene families, and then we use TASA to build the phylogeny, and then we put all thousands of gene trees into MAPS. So quickly give you 2 examples. Each of this is a gene trees with 3 taxa, A, B, and C. So the first step, we collapse impair logs because we only interested in the shear duplication event. You can see that in this example, all the B impair log have collapsed in the first step, and we don't have impair logs in the second example. And then in the second step, we sort the gene tree topologies, and then MAPS will identify if there's anything, any duplication on those trees. In the first one, we don't observe a gene duplication in here. And the second example, this is a gene duplication that causes specific gene tree topology. And then we've done this for every single gene tree that we got. So if there is a shear duplication event, we expect to observe this increase in the percentage of substrate that support the duplication at a certain node. And then if there's multiple independent duplication event, we do not expect to see that. And I just give you a quick comparison here, a shear one and multiple independent ones. So just give you a few examples from the empirical result from the from our study. Here is showing you the whole Osmondaceae shear 1 genome duplication event. And also in the Cyaphales, in the tree ferns, we do see 2 rounds of ancient genome duplication event. And also, we confirmed that with the first 2 genomes that we just got out last year in collaboration with Byway Lee at Cornell, using the Azola and Salvinia genomes. So first, we do the KS plot again. We do see there's polyploidy in there, ancient polyploidy in there. And then we also do our maps analyses. So the blue line here is the empirical result. The multiple gray line here is showing you tons of non-simulation that we have. So that's what you expect to observe with no ancient genome duplication event. So maps do infirm there's 1 round of genome duplication event shared by the genus Azola, and there's also another round of genome duplication event earlier shared by all the corn leptosporangia ferns. So since we have pretty solid genomes, what we can do is we can also do syntenic analyses. So syntenies, if you're not familiar with that, syntenies are the collinear blocks in the genome that are result from whole genome duplications. Since we got the genome, we can do that. We do the syntenies, and then we also pulled out the duplicate genes from the syntenic region, and then we also calculate KS for those duplicate genes. So what we found is we also seeing there's a whole genome duplication event shared by a genus Azola, and there's another corn leptosporangia whole genome duplication event. If you put this 2 results together, it's consistent. We also both phylogenomic analyses and the syntenic analyses showing you the same result. So just to summarize that, so across all the data that we got, we inferred 26 ancient genome duplication events across the fern phylogeny, including 1 in the ancestry of all the ferns. So going back to our question, so why do Homosperous fern have so many more chromosomes compared to flying plants? Well, 2 hypotheses: either they have more rounds, right, or they have similar round or even fewer rounds, but they just don't lose their chromosomes. So how do we test that? So the second thing is we need to estimate what's the frequency of ancient polyploidy in fern and angiosperm. So here gives you a toy example. How do we do that? So what we need to do is for each individual species, we need to count how many rounds of ancient genome duplication event that we had. So for example, for species E, we'll count from the root towards the tip how many rounds. So each of this red dot is a whole genome duplication event. For E, it will be 1, 2, and 3. So we another example could be C. We'll do from root to the tip will be 1, 2. So we do that for every single species. All we got from the fern phylogeny, we do that, and then for other lineages, flowering plants, gymnosperm, we do that from our current 1KP result, and then we summarize all the pattern here. So what we see in fern is, on average, fern species have 4 rounds of genome duplication event. Some have more. The lowest will be 2, so that's what we see in fern. In flying plants, surprisingly, the average round is also about fern. Some go up all the way to 6, and some only have 2 rounds. So if you put that together, they look pretty much similar. And if you compare to other groups, gymnosperms, lycophytes, we can see that there's a significant difference on the average ancient polyploidy between fern and gymnosperm, fern and lycophyte, but there's not significant difference between flying plants and ferns. So going back to our hypothesis, more round or similar round, well, apparently it supports our hypothesis number 2 that they do have really similar rounds of ancient genome duplication event. So just to summarize what we had, well, for first question, where did this occur? Across the fern phylogeny, we have 26 ancient genome duplication events, including 1 shared among all the neophytes. And we also have really clear result that the syntenic analysis and the phylogenomic analyses match up together from our first 2 fern genomes. So why do Homosperous fern have so many more chromosomes compared to flying plants? While Homosperous fern have similar rounds of ancient genome duplication event, but they just don't lose their chromosomes. Well, with that, I will acknowledge both Jim and Mike. They brought me to the world of plants. And I also need to thank the Barker Lab. Mike has been working on this project for a long time. Also thank our awesome undergrads, Brittany, undergrads, postdocs. Brittany and Paul, they always help me with They're just so knowledgeable. And I need to thank the funding source, NSF, the Galileo Circle from University of Arizona, and our EEB department. And then before I finish, I have, since Jim is not here, I will show you some picture. If you just walk in the room, you probably haven't seen this before. So this is Jim trying to demonstrate how bats get the nectar from flower when we are in Puerto Rico. And then I think this one, Jim is trying to find the red flowers in the bush. So if you know Jim's, that's a joke.
That's he's colorblind, yeah. So that's the best joke. It's fast. You say, there's a red bird. What is that?
All right. This is still from the Puerto Rico trip. Jim and Mike trying to sort out the rental car issue. And also, Jim is trying to teach Tia how to press the plant, or they're just keying out the fur, and I forgot what exactly that is. And this is from 3 years later. We are at the Tucson Pecos Island. I think Jim is ready to get some breakfast, probably finding another red flowers. And I don't know what he is complaining about. And then June and Mike teach us how to plant in the field. And if you know him, this is what he do in the field and also in the film hood.
So folks that don't know Jim very well, he had a fume hood in there, and you go to meet with Jim and work on a paper, and that's what he do. That's what he do. Yep, in his office without anyone knowing, I guess. So.
We joined.
Yeah, there you go.
And then I think this is he trying to make fun of Justin. So when I was putting a talk together, I'm really trying to sort of find a way to summarize how much impact that I got from both Jim and Mike. And I'll put it this way: if I can sort of rank any human being that I encounter in my life and sort of rank how many impact that I have from them, I think it will be my parents and then these two guys. Jim and Mike. Thank you so much.
Before we finish up here, we've got one more little talk to do. Now, when we first put this together, Jim was going to come out, and that was the plan, and that he was going to come out and we named this. This is Jim on one of these Bahamas trips, being Jim. And we call this questioning species and species complexes. This sort of encompasses, I think, anybody that sat in with Jim, he really asks you questions and asks you to question everything. And I thought this was the right way to phrase this. That really encompasses all the work that all of us have done with him over the years. But I really, really, really wanted to do this. I knew Amy Litt would definitely not accept this as a title. But we all went to Miami and got Hickeyed. What I really wanted to call this. Because we did, and when I was there, we had a lab t-shirt that we wore at Botany 2003 that said I got Hickeyed, and had an Isodes on it. And we got lots of weird questions in Alabama. When you especially left the Botany meeting. But anyway. And then just to show this, what I guess is Jim's picture he picked to send us for the meeting. And I think this really summarizes Jim pretty well. But Jim, in a very nurturing way. This is a nurturing photo. Like, don't be so. But because Jim couldn't be here because of his health, he sent a statement that he wanted us to read to everyone that's participating in the symposium. I'm going to put it up here on the board and I'll just read it out here. So that, sorry to interrupt these talks, but I love to irritate all of you any chance I get. So 3 quick things. First, let me say I'm truly sorry I cannot be with you today. My health just isn't up to long flights and hot weather. I would have been there if I was able. Thank you all for coming, and thank you, Mike and Melanie, for getting this going. Second, now I think I'm the luckiest botanist alive. In 35 years at Miami, I have been blessed with the greatest assemblage of PhD, master's, and undergrad research students anyone could possibly wish for. Seeing all of you accomplish so much across such a diverse arena of interest makes me humble, and not a feeling I'm familiar with. I didn't write this. I didn't edit it. I want you to know that you are all like family to me, and despite our occasional or many, Ethan. There we go. He wanted me to look at you. He said. Arguments, I love you all. You've made my career worthwhile, so thank you. And finally, I hope you'll take this opportunity to thank Mike Vincent. Without him as a botanical life partner, I could not have succeeded, and I suspect neither would many of you. I know how hard he worked with all of you. All of my success lies in great part to his constant support and encouragement. He is my brother. He is the Felix to my Oscar. Thank you from Jim. So let's give everybody I spent a couple of years in Jim's lab, and it was wonderful. Mike was wonderful. I am so happy that they're both memorialized with species out there, recognizing their names. Hopefully they never get synonymized. But yeah, I mean, like Lee, Jim was a huge impact on me and my career, and you know, the attention and thinking about diversity and variation, and just really questioning. I've never met someone who is so interested in challenging people on how they know what they know, right? And it is truly a unique experience to experience it. And it doesn't matter who he's talking to and what their expertise is. He's able to find that thing and sort of find that weak spot and challenge it. And it's a skill, and it's amazing. And he was a wonderful collaborator and advisor to me, and I think to many of us here. So, but thank you all for coming out to Tucson, and thank you to Mike for being our shadow advisor and our partner in crime there. So, thank you, everyone.
And before you go, I want to take a picture.
Oh, we got to take a picture. That's right.
The Hickeyeites.
The Hickeyeites assemble.